mute the handphone and keep silent. So, ladies and gentlemen, the regional workshop and biotechnology for food security and biodiversity conservation commence. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Honorable the Director of Research, Technology and Community Service, the Director of Simio Biotrop, Dr. Zulham Shah Imran, the speakers, moderators, and all the participants, distinguished guests, ladies and gentlemen. First of all, allow me to welcome all of you and sincere our gratitude to your presence in this opening ceremony of the regional workshop on biotechnology for food security and biodiversity conservation. I'm Alia and I will be your master of ceremony this morning. As our first item in the opening ceremony agenda, may we present the national anthem of Indonesia Raya. Please rise. And standing for the Simio colors. Please be seated. Presentation of Simeo Song.
It's time to open our eyes to a bright and promising future. We'll rise to the call of the times and create a stronger tomorrow. Let's start to make a difference, teaching everyone to learn through science, health, and technology. We will share our expertise by promoting knowledge and policies. We can build a better world. Let us take a stand for our visions of hope, making future plans for the people to grow in a life filled with happiness and green. Here at Simeo, building the future is our legacy. We'll achieve our goals through partnership, creating opportunities. By working hand in hand, we can accomplish all our dreams. For a better quality of life is now within our reach. Let us take a stand for our visions of hope, making future plans for the people to grow in a life filled with happiness and green. Here at Simeo, building the future is our legacy. Let us take a stand for our visions of hope, making future plans for the people to grow in a life filled with happiness and green. Here at Simeo, building the future is our legacy. Building the future is our legacy. At Simeo.
Thank you. So, ladies and gentlemen, our next agenda that will be report from our coordinator. So, for Dr. Romi Ardiamsyah, please welcome.
Yeah. Okay. So good morning, everyone. First of all, for the Excellency, for the Director General of Higher Education, Research and Technology, Professor Dr. Insinyur Nizam, and the Honorable Director of Samaya Biotrop, Dr. Zulham Saimdran, Deputy Director for Administration, Dr. Ferdinand, and for all the managers and head of section and head of the unit in the semi-biotrops and all the staffs. And for Honorable also, for all the speakers, all the resource persons, and for all the moderators, facilitators for these regional workshops, and also Honorable for all the participants that have made them available for today's activity. Assalamualaikum warahmatullahi wabarakatuh. So yeah, I am Romi Ardiamsyah. So I'm conducting as the coordinator for these regional workshops on biotechnology for food security and biodiversity conservation. So we know that biotechnology, it's actually not the really new things. It actually has been the technology is already used for several years, for even hundreds years ago. that we know. We call it like the classical biotechnology, but currently biotechnology is really rapid advanced technology right now. We know there are several advanced technology in the biotechnology such as genome editing, and also several use and several uses of the biotechnology. So the point is, the biotechnology is really rapid in the rapid developments. So the question is the regulation is also follows the development of the biotechnology. So this why the regional workshop on biotechnology is conducting today. But however, we know that we would like also to know what is the role of biotechnology for food security, and also what is the role of the biotechnology for the food, for the biodiversity conservation, what kind of regulations that is need to be obeyed for us, and what should we know about the biotechnology, about its biosafety, and also how about the use of the biotechnology products for the food safety itself. So this is the regional workshops for on the biotechnology for food security and biodiversity conservations, which also a talk series, a part of talk series for in the biodiversity engineering, which is a road to the conference, because Semel Biotrop will be conducting the international conference, the fourth international conference on tropical biology, which will be held on October 25 to 27th. So this is the kind of the activity for that ACTB, and hope you or the participants, even the research person, could also join in that international conference, tropical biology, that will be conducted in this year. So thank you very much for every participants. Thank you very much for every research persons. We have several participants actually from Indonesia, of course. Also, we have several participants from Philippines. We also have from Cambodia. We also have several participants from Malaysia. Even if I don't have misunderstood, there also come from the Algeria, and also from India also comes, also join for this activity. Hope we can share a lot of information for today regarding on the biotechnology, the role of biotechnology for food security and biodiversity conservation. And we got several inputs also, not only from the research persons, but also from the participants. That's maybe from me. Thank you very much for everyone, and Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Romi, for the remarks. So, ladies and gentlemen, our next agenda will be the remarks from our director, Simio Biotrop. For Dr. Zohar Hamsyah Imran, please welcome.
Yeah. Yeah, thank you, Alia. His Excellencies, Director General Higher Education, Research and Technology, Minister of Education, Culture, Research and Technology, Honorable Deputy Director for Administration, Dr. Ferdinand. He is beside me now. Resource person, the moderators and facilitator of regional workshop, Professor Dr. Bambang Prasetya. Yeah, he is head of the Indonesian Biosafety Commission of the genetically engineered product. Professor Dr. Antonio Suwanto, my colleague from IPB University. Ilmi Kurniawati, Policy Analysis, Directorate Conservation of the Species and Genetic Biodiversity, Indonesian Ministry of Environment and Forestry. Sufiati Dewi, and then Dr. Evelyn Bigchas, our former Deputy Director for a Program, and then Commissioner Aldrin Darilak, our GB member, and also Commission of the Higher Education, the Philippines. Dr. Suprianto, of course, yeah, my senior, affiliate scientist of the senior Biotrop. Yeah. And then also all the participants, yeah, We can see also we have the participant from Japan, yeah, my colleague, uh, Dr. Candidate Primagandi, I think, yeah. He attended this meeting, yeah, from Tokyo. Yeah, of course, yeah, we have the so many participants come from various countries, yeah, not only from the Asia, including from Africa, maybe it can be from other continent, yeah. Yeah, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Yeah, first of all, let us praise and thank to the presence of the Allah Almighty for the abundance of grace and the joy of all us can still gather in the place without any barrier and all in good health. Thank you to each and every one of you for being here today at this wonderful place. Yeah, yeah. Of course, yeah. We use the hybrid platform. Yeah. Before we get started, I would like to express my appreciation to the organizing committee who generously have and organize us make this workshop come true. Welcome to Simio Biotalk. Welcome to regional workshop on biotechnology for food security and biodiversity conservation. Yeah, two things. Yeah, we need to discuss a lot. Yeah, because yeah, we know biodiversity not only for conservation purposes, but also how to use for food security. Yeah, we know that Indonesia is a country with a very large source of the biodiversity. Therefore, there are challenges that will face in effort to empower biodiversity. The stability and preservation of the biodiversity are increasingly threatened by the increasing number in intensity of stressor and pressure. Therefore, several strategies are needed to face future challenges related to the biodiversity. Simio Biobotop as a regional center for tropical biology also play a role in the Safe Biodiversity program. Yeah, we know that our tagline, Safe Biodiversity from mountain to the ocean. Yeah. The loss of the biodiversity in Indonesia is around 72%, significant threat to the ecosystem and human well-being. An increase in population cause an increase in the amount of the food need. Yeah. To achieve the goal of the domestic food sovereignty, several steps can be taken, including by increasing food productivity with the application of the biotechnology. Biotechnology is a way out our solution to face the challenges of the threat of the world food crisis, including Indonesia. Yeah, we know that the genetic engineering of food crop with the biotechnology must be carried out and developed in order to anticipate the of the world food crisis, which is predicted to peak starting in the 2050. FAO predict that there will be an increase in food demand by as much as 60%, so that the world population does not sink into poverty hunger. Breeding variety of holding plants such as rice, corn, sugarcane, and wheat by utilizing biotechnology must continue to be carried out in the future. With the genetic engineering technology, breeder can develop new varieties that are resistant to the agricultural problem such as disease and pests, waterlogging, and salinity also drought, because of the now increasing of the climate crisis. By harnessing the potential of the biotechnology in food security and biodiversity conservation, we can address the growing global demand for food, reduce environmental impact, and preserve the Earth's rich biodiversity for the future generation. Senior Biotech role in the supporting the application of the biotechnology in Indonesia can be seen from its contribution in the research, among other, by conducting genetic engineering to obtain superior seed, identifying the cloning gene resistant to pest and disease, and applying plant tissue culture to provide superior seed. Ladies and gentlemen, through this activity, I do hope that we could discuss regulation policy and risk management of the biotechnology, then develops appropriate tropical biology system, which is one of the center mandates. I also expect that we could generate our, our weather network for biotechnology for food security and biodiversity conservation. Finally, I wish you all have fruitful, meaningful, and memorable workshop activity which have been decided especially for our need. I can already see big potential from the participant. I do hope that through this event, we all can learn to be a good and critical thinker. Once again, welcome and thank you very much. Thank you so much for attending this event. I wish all you participants and then good luck. Thank you.
Thank you, Dr. Zulham Shah Imran, for the remarks. So, ladies and gentlemen, we would invite Professor Nizam, the Director General of Higher Education, Research and Technology, that will be presented by video show. So, please.
Assalamualaikum warahmatullahi wabarakatuh. Semangat pagi, salam sehat, salam sejahtera untuk kita semua. Very good morning to all of you. Dr. Zulham Shah Imran, Director of Simeo Biotrop, all key note speakers and academia, as well as researchers joining this morning workshops. First of all, I would like to welcome and congratulate all of you for joining the regional workshop on biotechnology for food security and biodiversity conservations, hosted by Simeo Biotrop from 20th to 21st July 2023. This regional workshops, I think, is very timely, given the fact that now food security is very much high in the agenda of every nations. Also, biodiversity conservations. In regions, in ASEAN countries, we know that our regions is one of the region where the biodiversity is the highest in the world, both on land as well as in the ocean and the seas, on the seas. And therefore, the food security and biodiversity conservations is just two faces of the same coin. We have to, at the one side, on the one hand, assuring food security for our nations. On the other hand, we also have to conserve the biodiversity in order to secure our future sustainability. Therefore, the regional workshops, which address those two sides of the same resources, is very important. And through these workshops, I do hope that best practices from each country, from researchers from CMEO member countries, can be shared and discussed to find better solutions for the future. Food security is the foundations for sustainable development, because food security also will address the issues of poverty, the issues of low health, the issues of weaknesses of development. Therefore, the assuring the food security is should be our focus, especially using the advancement of biotechnology to improve productions, to improve the quality of food, as well as to increase the availability and access to better nutrition for everyone. With regard to biodiversity conservations, as I said earlier, as resource-rich countries in the Southeast Asian countries, biodiversity is something that we are endowed with plentiful. Therefore, conserving those biodiversity will be capital for our future. We will be indebted to our future generations if we could not conserve the biodiversity that we have been endowed with. So with that, I would like to once again thanks everyone for making this regional workshops a success, and wish you all this workshop brings good discussion and conclusions that can be further advanced in the future through stronger collaborations in research and development between CIMEO member countries. Wish you all the best. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you. So, ladies and gentlemen, we really put high importance on our event today. Therefore, to memorize our precious occasion, may we invite you to take a group picture. So, for the participant who join online, kindly turn on your cameras. So, we have the camera session. So kindly turn on your camera for the all participants who join online. Yeah, thank you. So we will have a photo session. Please for the committee. 1, 2, 3. For the next slide. 1, 2, 3. For the last slide. 1, 2, 3. Okay, thank you, ladies and gentlemen. So now we will have a coffee break until 9:25 AM, and we will start the session at 9:25 AM. Will be facilitated by Dr. Evelyn for Mindanao State University. So now we will have a coffee break session for the participant who join in CIMEO Biotrop. Kindly enjoy your snack. For the virtual participant, kindly enjoy your snack in your room. So please wait until 9:25 AM. And the committee will be share about CIMEO Biotrop video. So please, Mas Indra. For the participant who join in CIMEO Biotrop, we will have a photo group session. So please come to this stage. For ladies and gentlemen, please.
Our Earth is faced with environmental losses and damages, plant pests and diseases, breaking of the food chains, floods, and long droughts. Within our journey of more than 50 years, CIMEO Biotrop is committed to consistently and continuously maintain its contribution and supports in sustainably managing and conserving the biodiversity and environment in Southeast Asia. We have great dreams, strong hopes, knowledge and capabilities to fulfill our vision. We have prominent extraordinary expertise to be the legacy for the future generation.
Regional Center for Tropical Biology is one of 26 regional centers under the Southeast Asian Ministers of Education Organization, SEMIO, located in Bogor, West Java, Indonesia. SEMIO Biotrop, that was established on the 6th of February 1968, aims to promote regional cooperation to address tropical biology issues in the Southeast Asia. Governing Board is the highest body in the center's organizational structure. The Rector of IPB University has a privilege to serve as SEMIO Biotrop Governing Board member from Indonesia. Tropical biology issues develop dynamically through three main mandates, namely research, trainings, and information dissemination. SEMIO Biotrop conducts its programs and activities to contribute significantly in addressing those issues. Program on School Fruit Garden for Education, Production, Genetic Conservation and Entrepreneurship in Agriculture Vocational Senior High Schools in Indonesia, SMARTBE, and Program of SEMIO Biotrop to support the vocational schools revitalization. SEMIO Biotrop is supported by prominent experts in implementing its programs and activities. Products as the outputs of SEMIO Biotrop's program implementation. SEMIO Biotrops its international networks, linkages, and various beneficiaries. SEMIO Biotrops is equipped with quality facilities in conducting its activities. We place our hearts for SEMIO Biotrop.
Our Earth is faced with environmental losses and damages, plant pests and diseases, breaking of the food chains, floods, and long droughts. Within our journey of more than 50 years, SEMIO Biotrop is committed to consistently and continuously maintain its contribution and supports in sustainably managing and conserving the biodiversity and environment in Southeast Asia. We have great dreams, strong hopes, knowledge and capabilities to fulfill our vision. We have prominent extraordinary expertise to be the legacy for the future generation.
Regional Center for Tropical Biology is one of 26 regional centers under the Southeast Asian Ministers of Education Organization, SEMIO, located in Bogor, West Java, Indonesia. SEMIO Biotrop, that was established on the 6th of February 1968, aims to promote regional cooperation to address tropical biology issues in the Southeast Asia. Governing Board is the highest body in the center's organizational structure. The Rector of IPB University has a privilege to serve as SEMIO Biotrop Governing Board member from Indonesia. Tropical biology issues develop dynamically through three main mandates, namely research, trainings, and information dissemination. Simio Biotrop conducts its programs and activities to contribute significantly in addressing those issues. Program on School Fruit Garden for Education, Production, Genetic Conservation and Entrepreneurship in Agriculture Vocational Senior High Schools in Indonesia, SmartBee, and Program of Simio Biotrop to support the vocational schools revitalization. Simio Biotrop is supported by prominent experts in implementing its programs and activities. Products as the outputs of Simio Biotrop's program implementation. Simio Biotrops its international networks, linkages, and various beneficiaries. Simio Biotrops is equipped with quality facilities in conducting its activities. We place our hearts for Simio Biotrop.
Greetings from Simio Biotech. We will take you to Biotech campus, located at Jalan Raya Tajun, Bogor, West Java, Indonesia.
As the community learning park, Simio Biotech is equipped with 14 research laboratories, as well as greenhouses and research fields.
to Siamio Biotrop. We know that over 50 years, Siamio Biotrop has contributed to address tropical biodiversity issues in the Southeast Asia and beyond through research, capacity building development, and publication. We envision to be a leading center in promoting and enhancing the real values to tropical biology in Southeast Asia.
Hydroponic and aquaponic units.
Tissue culture laboratory. Animal farming culture.
Okay, ladies and gentlemen, time show 9:25 EM. We will start our session. So we will invite our moderator, Dr. Evelyn V. Bikas from Mindanao State University, Marawi City, Philippines. Good morning, Bu Evelyn.
Hello, thank you, Miss Alia.
Okay.
Yeah, okay.
Okay, so please, Bu Evelyn.
My sound is okay.
Okay. Okay, your sound so clear. So please welcome Bu Evelyn. The time is yours.
Thank you. Thank you, Miss Alia. Government of Indonesia, high officials, Biotech Director, Dr. Zulham Shah, and Deputy Director for Administration, Dr. Ferdinand, distinguished speakers from the Indonesia Biosafety Commission for Genetically Engineered Product, the IPB University, from the Biosafety Secretariat of the Convention on Biological Diversity, and from the Indonesia Ministry of Environment and Forestry, partners from the Ministry of Environment and Forestry, Republic of Indonesia, National Agency for Drug and Food Control, Republic of Indonesia, International Rice Research Institute, the Philippines, International Service for the Acquisition of Agri-Biotech Applications, the ASEAN Risk Assessment Center for Food Safety, from the Convention on Biological Diversity Secretariat, from the Southeast ASEAN Center for Graduate Study and Research in Agriculture, or SIAMIO Siarca, the Philippines, from IPB University, Republic of Indonesia, from Mindanao State University, Marawi City, Philippines, and the Indonesian Biotechnology Information Center. Valued participants, ladies and gentlemen, good morning, and thank you for gracing this event with your presence. I am Evelyn Bikas, and I am so honored and privileged to be your moderator this morning for this morning session. I would like to welcome you all to this blended webinar of the regional workshop on biotechnology for food security and biodiversity conservation, with the theme that focuses on the role of biotechnology and in food security and biodiversity conservation, including its risk management and regulation. This activity aims to obtain input and information from the stakeholders and resource persons in identifying the role of biotechnology in food security and biodiversity conservation, information on the progress or updates on biotechnology for food security and biodiversity conservation, information on the regulations and policies in utilizing biotechnology for food security and biodiversity conservation, and information on risk management for biotechnology products. I hope that everyone will be delighted to listen and learn new things and updates from our seasoned speakers on issues that really concerned us as human beings, particularly on the topics on biotechnology and food security, biotechnology and biodiversity conservation, effective participation in the Biosafety Clearinghouse of the Cartagena Protocol on Biosafety, and implementation of Biosafety Clearinghouse, the Indonesian experience. However, before we proceed any further, I would like to remind everyone that our esteemed speakers are given 30 minutes to present their papers, and there will be another 60 minutes for all four of them for the Q&A, or the question and answer, at the end of the session. Towards the end of their 30 minutes, the speakers will be prompted or reminded through a ringing bell or whatever that their time is almost over and they need to wrap up. After all our resource speakers have presented, the participants will be given their chances to shoot their questions, clarifications on the papers presented. We would also like to give our resource speakers the chance to give suggestions in relation to Biotrope's programs and activities, and for future research endeavors in biotechnology in the Southeast Asian region and beyond. Our topics for this morning to be presented by the following. Topic 1 is on the role of biotechnology in food security, to be presented by Professor Dr. Bambang Prasetya. Topic number 2, the role of biotechnology in biodiversity conservation, to be presented by Professor Dr. Antonio Suwanto. Topic number 3, on the building capacity for effective participation in the Biosafety Clearinghouse of the Cartagena Protocol on Biodiversity, to be presented by Mr. Wadsanai Mandiveni. And last but not the least, topic 4 for this morning, on the implementation of Biosafety Clearinghouse, the Indonesian experience. Okay, ladies and gentlemen, our first speaker for this morning is Professor Dr. Bambang Prasetya, who hails from Tangerang Selatan, and his present position, current position, and tasks include the following. He is a principal researcher or research professor at Research Center for Testing, Technology, and Standards of the National Agency for Research and Innovation, or the BRIN. He is the chairman of the National Biosafety Committee for 2022 to 2026, assigned by the President of the Republic of Indonesia. He is the main commissioner at the PT Quantum HRM International. He chairs the ISO Technical Committee 311 of the National SNI TC 13 to 14 BSN. He is also a member of the National Quality Award SNI Award Committee. He finished his bachelor's degree from Bogor Agricultural University. I will not mention when was that. He finished his master's from George August University, Göttingen, Germany. And his PhD from the same university, George August University, Göttingen, Germany. He did his postdoc at Kyoto University in Japan in 1995. And he did a national resilience course, LEMHANAS, in 2001 and national administration course in 2019. His occupational experience include being, so actually there are many, so I'll just mention a few if you spare me, Professor Bambang. He is a researcher at Indonesian Institute of Science, or the LIPI. And he's the chairman of the National Committee on International Standards of IEC and Codex Alimentarius. And for his professional membership, it includes being the chair of Advisory Board KBI and AFOB, currently from 2020 and until now to 2023. And he's a member of Indonesia Academic of Science, starting 2019 to 2023. He's the project leader of the RIIM, or the Research Innovation for Indonesia Moving Forward, Development of Traceability Standard of Palm Oil from 2021 to 2023. His scientific publications, in total, 235, 48 of which are internationally indexed in the International Index Corpus, 12, and research 358, and then the Index Google Scholars 17, and many others. So he has 6 patents in Indonesia and 1 patent international. He is also the ISO assessor, the current one. So for ISO 17025 for management system for laboratory, ISO IEC 17024 assessor SDM, international certification for human capital management based SNI ISO 17024. And he also is a recipient of the President Award Karya Pembangunan in 2003 and 2007, and in BRIN, awardee also of the BRIN Sarwono Award in 2022. So, ladies and gentlemen, please help me welcome Professor Bambang and his presentation on the role of biotechnology in food security. Professor Bambang, the time is yours.
Thank you very much, Ms. Evelyn Bikas, for the kind of introduction. Before I start, I would like to share my presentation. Want to check the picture and my voice is okay?
Yes, sir.
Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Distinguished invited speaker, and special here, Mr. Julham Shah, as a director of Biotechnology, and other invited speaker, and ladies and gentlemen, all participants, either in offline or in online. It is very happy for me to have the opportunity to share with you all about the role of biotechnology and on the food security. Just to make okay. From the organizing committee asked to me for the presentation number 2 until number 7. It is quite big tema in a short time. I think I will running quickly. But before I enter the real topic that the organizing committee asked for me, I would like to share about the global challenge because I'm here as the first speaker. I think it's better to tell you about the platform and our thinking. I would like first the global challenging and the risk mitigation. As we know all, ladies and gentlemen, that the Global Risk Report 2023 report that what is our concern in the food security is very related to the trends in the 10 years later. For example, the climate change and the extreme weather and the biodiversity loss and other, for example, the natural damage and also the natural resource crisis is very important with our topic today's related with biodiversity and also the food security. So maybe our talk will related with this kind of topic is very much significantly with the biotechnology role in the future. As already you all may be aware about the climate change is influenced by the global warming, the global warming increase the temperature and also the emission from the gases, CO2 and also methane. But what we be optimistic in the future because there are also policy which very effective to reduce the global warming. For example, in the slide, I will show you about the scenario, for example, the current policy and the better scenario and what happened when we know scenario about the emission of the gases. This is very important to us to know about this situation because, ladies and gentlemen, the climate change influence in our food system. For example, like in this picture, the impact in agriculture will decrease our yield, productivity, increase the nutritive quality, the nutrition quality, and also maybe the cerebral disease will attack to our plant. And I think it is very interactive between increasing flooding, increasing the salination, and many other things we can also address by biotechnology in the near future. Based on the research until now, we can observe the research result. Many research results already successful to address this kind of the problem. And this slide, the drought and also water stress. This is very often now because the climate change, the periodical of the weather is not predictable. This is bring difficulty from the farmer, especially in our country, like in Java, in Pacitan, and south of Indonesia, Java Island, and also in Nusa Tenggara. Is very difficult to predict of the good planting of agriculture plant like maize, for example, that I learned from the Pacitan. So the biotechnology role is very important because when our result of the biotechnology research, many variety can address to be resistant to drought, for example, resistant for the substandard, unfertilized of the soil, for example, soil with high salinity, soil with very acid in the peatland, for example. This already many researchers in biotechnology takes role to address kind of the global risk, drought, or and also the flooding. In this slide, I combine between the deforestation that we have available land is decreasing by time to time. In the other side, the population also increase very high, starting from 1950. It is also the same time, 1915 also the Watson, the double helix also found in the biotechnology. This is actually a parallel is very significant connection and also the problem. Thank the God that our scientists also many effort to address involving the population by throwing the technology, including the biotechnology. And another cases, especially in Indonesia, maybe also in another country, we still have the food security and number and also the hunger. This is the map of the global related to the hunger, the potential in hunger, and also within malnutrition. I mean, in the global, for example, the vitamin A deficiency in the global map. For example, in Indonesia, also in the cases at the zinc and iron bring to the we call it the stunting. This is also a serious problem in several locations in Indonesia. So we have to address kind of this biofortification plays very important role because we can put a certain nutrition in the plant. For example, in the rice, we know all that golden rice is iron, zinc, and some research in Indonesia also concentrate with kind of cassava enriched by iron and also provitamin A to make a variety mix rich in the vitamin A. This is the role of a biotechnology to facing of several problems in the world and also in our country. I skipped this slide. Talking about because the global climate change also influence the biodiversity loss. For example, here in the picture, maybe later on my colleague Professor Antoni Suwanto will talk about the biodiversity loss. I skipped this slide, but I just put from the roadmap of Indonesian action plan on the biodiversity that this is our life in the local people, that biodiversity bring several potential for his life, for the food, for the protection, from the recreation. This come the income from the local people. And then, ladies and gentlemen, I would like also show this interaction between biotechnology for the sustainable biodiversity and also food security. In terms of the biotechnology can reduction from the, for example, over exploitation by creating a species certain with a high economical. There are level of the biodiversity and genetic and species and ecosystem, and also we can combine what kind of the problem we can address by, for example, by tissue culture. I think the biotrope is very experienced. We propagation, cloning, artificial insemination in terms of the animal science and bioregion in terms of soil and remediation, and also genome editing. The last technology is very endorsed by government and by scientists to make a more research and more quickly result. And then I also asked to just overview the history of the biotechnology, but it is, I think I can very quick to review this one. In fact, from the slide we can see that the milestone of the biotechnology starting from the molecular biology, and then what is important for us actually the parallel in line with the research activity, also the development of equipment. So for example, in the molecular biology, there are many equipment developed with precision and now with high capacity and precision. Therefore, lately, the research on the biotechnology growing very fast. When we're talking about the breeding, I think my colleagues very aware about the milestone in plant breeding. For example, from the Mendel theory, and then by mutation, by recombinant protein, recombinant, and lately by genome editing is very promising technology, can provide the technology alternative from the plant breeding. I skip this slide because, and then in overview of the design of the new variety, we can select the new variety by, for example, in the DNA level. And also we can also select in the plant level, for example, phenotyping, crop modeling, and in term DNA level, mapping population marker assisted selection, target population. And then when we're talking about new plant varieties, we can also at the DNA level and also the plant level. For example, at plant level, we can conventional breeding is already done by ordinary people. There are many area in Indonesia found a new variety by self breeding, cross breeding, and then metabolic design, mutagenesis, and so on. When we're talking about the DNA level, we can know the product GMO is very huge in Indonesia also, that it can transfer and ciskinase, transkinase, and plastic transformation, and also many technology, including genome editing, transposon, gene silencing transposon, and also mutagenesis. We know very well about this. And then when about the new tools, the trend of the not only the genome editing, but many alternatives we can develop in terms of the setting of technology for a certain variety. For example, starting by synthetic biology, protein engineering, and then gene silencing and editing, genome editing, and after that, bring to the small greenhouse and you can try to make a plan in the small greenhouse, and then we can try in the field, and then recording regulation, so we can also apply certain variety can be released in a certain area. Ladies and gentlemen, now I would like to share about the role of biotechnology in the food security. First of all, the goal of biotechnology actually in three categories. First is yield, and secondly, nutritional quality, and also the productivity sustainability. For example, the yield, improving photosynthesis, increasing the size and harvesting part, and improving the use and the movement of the nutrient within plant, altering, growing in season, and then improving of the digestibility of biomass, for example, for the feed. And then when we're talking about the nutritional quality, increasing vitamin, increasing mineral content, increasing protein or quality also, decreasing negative effect, for example, toxic compound, increasing antioxidant, producing healthy omega-3 fatty acid, for example, in oilseed. And when we're talking about the productivity, sustainability, we can also improve the nutrient use, improving plant water use, improving phosphorus uptake of use, because phosphor fertilizer based on phosphor is cannot synthesize in the plant. This is different with the nitrogen. And then we also improving stress, especially when we talking about the farming in coastal area. For example, I have experience in Gunung Kidul. We can also plant a certain plant in the sheltered area with high shelter. Kemudian also improving hot stress tolerance. I put here the journal publication from the European, because the European in terms of GMO is rather different with another country. But one publication here talking about the role of the new plant breeding technology for food security and sustainable agriculture production. This is very a good overview about the benefit and the perspective on the GMO in terms of perspective on the European country, for example, here. And here talking about the benefit, the same paper talking about what is actually the benefit of GM crop in terms of how high the yield can be increased, how about the pesticide quantity can reduce so much, and then farm profit, and then the life side, the impact of the BT cotton adoption in India, for example. This maybe colleagues from India know better about this. And from the ISA, I think it is a good overview about the benefit, the contribution of crop to food security, and sustainability and the climate change. There are data about the increased crop productivity. For example, they are very quantitative from 1996 to 2016, around 20 years. Yeah. And then conserve biodiversity and how they can provide a better environmental and reduce, it is very important for related with global warming to reduce CO2 emission, for example. How high actually the biotechnology can reduce the CO2, for example, and help the smallholder farmer in certain country. This recorded in 20 years report from the ISA. This is very important for us to know about this role of the biotechnology. And I put this from the St. Louis last month, not last month, several months ago. I quite interesting about this picture because in Indonesia, like now, the scarcity of fertilizer is become in the our farming. So because this is very related with distribution, related with price, related with subsidized distribution, and so on. So how is the biotechnology play a role in this scarcity of the fertilizer? For example, we already know that the lithium can uptake the nitrogen from the air. But through biotechnology, this is not lithium only, but another variety we can also design to make the function as lithium to fixing of the nitrogen from the air. But not only the nitrogen, instead it is actually representative of need of urea, urea NPK. We need fertilizer based on the nitrogen, also the phosphorus. We can also design how the plant can manage the phosphorus, the phosphor. This is a kind of technology very promising with us because it's very, very close to our need. And then, ladies and gentlemen, when we're talking about the soil degradation, soil substandard, unfertilized, through certain microbe designed by certain genetic, we can place for example, bioremediation, phytoremediation, and also to uptake certain chemical, for example, heavy metal, but also in certain area very exceed the structure not so good, and then the biodiversity of microbe, which is very important for the soil science, for the soil biodiversity, for the soil properties. So the microbe we can inject in certain purpose to improve our quality. This is the microbiology. And talking about the yield productivity, several countries through certain report is confirmed that GMO crop increase the productivity and later on also income of the smallholder farm is also increase the income. Another data I also propose here, just summarize that the quantitative benefit of the GMO crop, for example, to increase of yield, labor saving, fertilizer saving, increase of the income, saving the pesticide, fungicide, saving another mean of production, for example, and also the increase the biodiversity in the soil. Type side is table I put from certain literature, and also here in picture from the is original Indonesian research, and also some GMO crop plant in east of the Nusa Tenggara. Ladies and gentlemen, I also asked to prepare the current progress in development biotechnology for the product output. I starting by this is of the mapping of the role of the biotechnology, animal, plant, microbe, environmental, and health. Every sector, every part can divide several topics for student maybe know better about the certain thema related with microbe, biotechnology, environmental biotechnology, and health medicine plan also. This is the mapping, I think, to know about the how is the scope biotechnology which can play a role in certain aspect of the food security. Position right now, ladies and gentlemen, the plan. This is the map of the world, and this is the number how big the plan of the because the starting sugarcane. But right now we have already 10 GMO is already planting following the sugarcane. And here's another data, the data source about the aerial in the several countries. Of course, it is United States, Brazil, Argentina is very last, the planting of the GM crop. In this slide, I will like to show you about the commercialize of the GMO in the world, which traits role in this. For example, herbicide tolerant and nutritional improvement, disease resistant of abiotic stress, and what kind of the GM crop state here, just to make an overview of GM crop. And in total, I scanned from the literature 2020, this GM trait is already around 46 GM trait list, but already commercials around X GM trait. It is, I think it is quite good to report. And when we're talking about the biotic and abiotic stress, there are table talking about
Me, Professor Bambang,
what kind of yes.
2 minutes
Okay.
more to wrap up your
How
presentation.
many minutes?
you.
Yes. How many minutes?
2 minutes more.
Oh, 2 minutes. Okay. I would like this about the genome editing. I would like to continue because the time. Yeah, I would like the of the lecture. Now Indonesian actually this is the mapping of the genome editing regulation. This is very important because in the beginning also known about role of genome editing, and Indonesian and yellow color is actually in the discussion. But now we can also already accepted the genome editing product. We can also approve because SDN1 and SDN2 we accept some from the GM regulation. But official regulation from the ministry not yet available because the KKH also have the responsibility to select whether it is GMO or not. This is important information from the GM editing. And then I continue to pros contra. I think not so important. You can read by my paper. I would like just information, but how to guarantee to assure that from the GM product we following aspect. For example, for biotic and the biorisk management and also conformity assessment. And I continue because the time biotic and it is the halal issue because we have already decided that halal also involved in the GM crop. And I think in several aspects we can also answer about the position of the pros and cons of the GMO. And let's just to show you about the seeing is believing. When we don't know about the GMO, so when you visit in the Pekan Nasional, the view of the National Week Agriculture last month, so we'll see about the potential of the GMO. This is the product. I put the picture from the Pekan Nasional, and this very nice GM crop that's arranged by the productivity compared with hybrid, compared with conventional, compared with other product. I think this is very important. And the people, participant, very enthusiastic to follow the technology transfer, knowledge sharing in this forum. And I think the regulation, I think this is the same. It is the basis of the committee. I would like to show you that our last slide maybe tell you that the business process how to apply GM crop and also GM genome editing is as follow. We can see also in our bioclearing website that telling about this process. And I think what is important conclusion that we have some about our assessment about the biosafety. For example, we have already 49 products here and related to biosafety for soybean, corn, and so on, and also recommendation for the 20 product for the vaccine, also 6 crop and for environmental biosafety, and from the feed biosafety 6 product. And it is the list our achievement for the biosafety committee, the recommendation of biosafety, and I think I skip. And last slide that recommendation for the biodiversity standard committee and the decision committee, we can formulation of the standard for all the world. So I think the Biotropic can share what kind of idea for the biodiversity standard we can propose the international standard. This is my last slide, and come to the conclusion that biotechnology is very important to face several problems related with global warming. And the result of technology in biotechnology is already very advanced now. The regulation in Indonesia is already prepared. Now we looking forward the result from the our college to bring the result for the technology to address the food security and food safety. Thank you very much for your kind attention. Sorry about the time.
Thank you very much, Professor Bambang, for this life-enriching information that you have just shared with us. Thank you for showing us the importance of biotechnology in food security. We do understand that food security is one of the SDGs, one of the 17 SDGs. Anyway, for those who would like further clarifications, later on you will have your chances during the Q&A. Thank you very much.
Thank you very much.
So, without any delay, our second speaker is another professor, Professor Antonio Suwanto, who is working currently at IPB University in Bogor, and he also hails from Bogor, Indonesia. He finished his bachelor's degree in agricultural technology at IPB University, his master's degree in microbial genetics at the University of Illinois, Urbana, USA, his doctoral degree in microbial genetics from the same University of Illinois, Urbana, USA, and his experiences and achievements include the following. He is a member of the Indonesian Technical Committee for Environmental Safety for Genetically Modified Microorganisms. He is a member of the Indonesian Academy of Sciences, a former Indonesian ambassador for American Society for Microbiology, first to report more than 1 chromosomes in prokaryotes. He is an awardee from a number of international research grants such as the Rockefeller Foundation, the IFS, Turai, and Kehati. His publications and products relate to enzyme biotechnology, Tempe microbiome, and probiotic applications. Without further delay, ladies and gentlemen, please help me welcome Professor Antonio Suwanto and his presentation on the topic on the role of biotechnology in biodiversity conservation. Professor Antonio, the time is yours. The next 30 minutes are all yours.
Okay,
yes,
now
sir.
Thank you, Dr. Evelyn, for your kind introduction. I would like to share my slide, okay? Okay, this is the title, that I would like to convey for this meetings for today meeting. So biotechnology for biodiversity conservation. So I will limit mostly on the some example. In fact, this is a good real example for biodiversity conservation. Okay, first, if you look at here, we look at all of the system in nature. There are so many diversity, of course. We can see so many things, including human here. But if we look at this all, actually, even though they are looks different, plant, animal, bacteria, everything, they have many similarity. If you go to the cellular level, and one of that is that their language, all of them is they're written down as AGCT, which is actually in the DNA. Whether this is actually corn, maize, orange, or maybe snake, human, all you have AGCT. And in fact, we can see here that we are related to each other. Of course, human to human is very related. You can see here, we share almost 100% our DNA between each other in terms of DNA sequence. We have with the chimpanzee slightly lower. And then with dogs, we still have similarity, 84%. With fruit flies, that one is 47%. Even with the grapes, the wine grapes, we still share similarity. So in fact, the whole system actually relate to each other. So if we talk about biodiversity, actually we are variants to each other. We are variant to another human, but also we are variant from primate. And we are variant also from other living system here, actually. That's why when we talk about biodiversity, we should respect every forms of life because they have their own role and then they are complementing each other. So we cannot just do by ourselves. This is actually the most essential concept because we comes from almost the same basis at the cellular level. And all of that share the same mechanism, how to convey the information molecule here. So how we make a protein, enzyme, or organism depend on the recipe. The recipe is written down as a DNA. This is a molecule, a polymer, and this polymer becomes protein through these mechanisms. And all of these mechanisms shared by all of the living system, whether this is actually a banana tree or dog or human will follow this one. That's why this is, they call it central dogma of molecular biology. That's why if we look at nature, they looks different, but actually all share similar thing, just like this, exactly. That's why you call it central dogma. From bacteria, plant to human, we have this type of thing. So the information stored as a DNA molecule, and then this will be processed to be a protein, which is become enzyme or other things that's compose organism. So in this case, for example, we can change the DNA through many types, whether they are naturally muted, change the DNA, and they can become different variety, or we mix it faster. The fast one usually through sexual reproduction. Sexual reproduction is the fast way to make recombination of the DNA, just like ourselves, human, our son or daughter, they're just very different from their parents, only in one generation. That's the power of sex. So through sex, you can see here, broccoli, you have cauliflower, you have cabbage from the same ancestor, which is wild cabbage here. So for example, at the year around 1500s here, there is a broccoli, but before that, there is no broccoli. Even during the era of cabbage here, we might not see broccoli or cauliflower because this is developed later on. Moreover, you can see now broccoli that have different type of, no, this is actually cauliflower with different types of color. So this is maybe, for example, if you have a chance to see in the supermarket, in some countries like Japan, you can see already some cauliflower with violet or different color. That is because of the sex. So through sex, human has been doing this kind of gene transaction, actually. How we change the genetic level, that is very fast. So we can understand this through the genetic study. For example, this one already initiated by Gregor Mendel since 1800 in here. So we already understand, or we learn this one since near high school, for example, how we can see the segregation of traits relate to the crossing or breeding. We can have the yellow seed or the green seed, but with the dwarf or the tall trees like this, and then we can cross it. So we can have the tall trees, but with the green color seed, for example. This is through breeding. So conventional breeding is actually a random genetic modification because we don't know the result. We can calculate actually, and we can see that this is inherited, but we have no control on the result in that case, or partially. So during this time, we can create many types of agricultural product. For example, here we can see that agricultural product like this is the result of genetic modification. How they modify? Modification is just through crossing, just through breeding, just sex in this case. And of course, selection, because human have some preference. For example, you need the sweet one, or less seed, or even seedless, something like this one. So that we can have a high yield and then resistant to certain disease or something. This is actually the result of genetic modification. So genetic modification is nothing new. In fact, also the blue, the ladies in blue here is also the result of genetic modification from her parents. But human actually not just only rely on what nature gives normally. In some cases, they found like fruit like this, watermelon. This watermelon without seed is nicer to enjoy for some, because we don't like to throw only just seed, for example. That may be inconvenient. So then we try to have fruit without seed. But this one, if we look at in nature, this is actually a terminal, meaning there is no more progeny. So they cannot survive. So finally, people want to understand why this fruit has no seed. Even though this one in nature is abnormal, but then we like it. Then we know this one is triploid. And this one is actually many of the plant with no seed are polyploid. So we induce it using chemical. Then we got it. And there is no problem related with this one in terms of safety or something. We just eat it. So chemical has been used. For example, in this creation of watermelon. In addition to that, we also use physical. Many of our grains like rice, maize, and also soybean like this was improved through genetic modification, employing physical. For example, this one, in this case, radiation. You just radiate all of this seed and then try to grow it and then expect to see whether they can have some genetic changes that make them resistant to disease or have higher yield. So we use physical, not just chemical. Also sex. This is actually, but then later on when we understand about DNA, we have known that DNA is actually convey genetic material, then we go also to employ these techniques, how we use this one. So for example, in the corn, for example, here we have a borer, which is actually the larvae that eat the corn, and this one actually very difficult to control. And of course, this create loss in the agriculture. So we can look at the natural enemy of this type of pests. Many of the insects has natural enemy, which is in this case, if they eat it or they encounter it, they can die. So for example, Bacillus thuringiensis is belong to that. So when insects ingest this bacterium, they will release the protein toxin, and the protein toxin will make the larvae die. But they need a condition. They should be alkaline. So the gut must be alkaline, so that makes this crystal protein disintegrated into their subunit and then finally inserted in the cell membrane of their intestine, and this become leaky and make the larvae die. So this is actually very specific, because if we ingest this, and actually we ingest bacteria like Bacillus thuringiensis almost every day in grain, in vegetable like that, and there is nothing with us, no reaction or anything, because this protein just become protein like the other protein. They cannot become a toxin because they cannot be disintegrated into this, since our stomach is acid, for example. So this is very ideal because this Bacillus thuringiensis very specific. So we can use it only to kill certain larvae, not killing human, animal, or other system. So that's why this type of bacteria has been used for many years as a organic pesticide, which is like actually very environmentally friendly. That's why organic farming using this. So with DNA technology, we can just take the gene from the bacteria that encode the protein toxin. In this case, it's cry gene, and then inserted this into the corn. If we insert this into the corn, then the corn will express the cry gene, and then they make protein, the toxin protein that is specific for this larvae. And this is, we call it the transgenic corn or genetically engineered corn, since this is getting gene from other organism. If we can make this bacterium do the sex, okay, make sex with the corn, we will do it. But since we cannot make sexual activity between bacteria and corn, then we cannot do anything. But with DNA technology, we can take the DNA and then the DNA will be expressed. This is because of the central dogma. Because the corn now has the recipe. And because they have a recipe, they make the toxin. Because they make the toxin, then they can become corn that can be protected. But this one use DNA technology. So then the result we can see, for example, this is the regular corn. They have a lot of damage. And then this is the normal corn. The same thing for soybean, for example. The soybean damaged heavily because of the infection from the larvae, but the left one is actually the one that is that is a transgenic soybean. This is actually has some little hole if you observe it, because the larvae need to eat it, to ingest it in order to get poison. If the larvae only just crawling here, they just stay okay. So biotechnology could help to reduce more harmful pesticide. They help the farmer from doing a lot of work by spraying or something. If you can see here, they have a lot of pesticide, synthetic pesticide, including insecticide used by the farmer to spray. So with the transgenic BT corn or BT other potato or something, they can reduce more harmful pesticide and provide better life quality for the farmer. That is an example, and so many examples from there: soybean, BT soy, glyphosate, everything. But I would like to tell you more here now about how the biotechnology can save some plant that is almost extinct. So this is how biotechnology can actually preserve biodiversity. So this is actually chestnut. Maybe some of you know about this already. This is not non-tropical plant actually, but the chestnut is like this. So you can buy it in a reedy form like this one. This is chestnut. That is no shell. This is with the shell, and this is the fruit itself. Actually, the tree is a beautiful tree. Especially this is actually for the American that thinks that this one is a very beautiful tree. And so you can see the canopy is very wide, and then the tree tall, so you can have a like picnic under the tree, for example. So this is a American like this one. But unfortunately, in the 1915, around 1900, the American forest that is mainly, especially in the Northeast USA, this one consists of chestnut tree. But then they got infected by fungus here. They just have a blight infected chestnut. Almost they gone now. So if you see it, only few left already, the chestnut until now. What happened here is because they infected here by the fungus called Cryphonectria parasitica. This is caused, says, chestnut blight. So the chestnut blight is a fungus, and this kill the trees actually. So then people try to make a crossing to the more resistant one. The more resistant trees is coming from China. The Chinese chestnut is more resistant, but then the posture, the stature of the architecture of the trees is shorter and not so wide, the canopy, not as beautiful as the American one, but they are resistant. So they try to make this one crossing to the Chinese chestnut. But when they got the resistant one, they always got the short one, almost like the dwarf type. So they don't like it actually. So what they wanted, the American chestnut that is very big and a beautiful tree. And when we go further, why this is poison? This chestnut, the fungus can poison the trees, the chestnut trees. You can see here that they have oxalic acid produced by the fungus. The fungus actually poison the trees because of oxalic acid. And then people also understand that in wheat, they have enzyme called oxalate oxidase. This is naturally in wheat. In wheat, then if there is any oxalate here, then there will be degraded into carbon dioxide and hydrogen peroxide, that which is finally become water. So people clone this gene from wheat and try to introduce this to chestnut. So whether this one will help and make resistant to chestnut blight, because there is no way to save the chestnut. So they take this gene from wheat and introduce this to chestnut. This is from wheat, but chestnut don't have the oxalate oxidase, but wheat has this one. And the fungus that infect chestnut, they produce oxalate. So this is the result here. If you look at the Chinese chestnut infected by the same pathogen, and this is the normal American chestnut, taller, bigger here, and the Chinese chestnut, this is resistant to chestnut blight, but they are short in stature. But if you give the OXO gene, oxalate oxidase, you can see this is like normal, big also, the big tree, tall, not like the Chinese chestnut. This is the positive control. This is negative control, actually, the normal chestnut. So it worked. But then people think, what happened in the environment if we put it into the forest, this GMO chestnut? So then they have to test it, whether they will influence forest plant germination, or maybe they influence mycorrhiza colonization. So they tested many things just to answer whether when we have introduced this GMO chestnut will be useful or give the good result. So one of the tests is how actually this litter, the tree litter, impact on the tadpole, because tadpole normally they also eat tree litter. So in this case, the tree litter from chestnut actually eaten by this tadpole. And here I give you just a cartoon that represent the growth of the tadpole. The big one is the high growth. So if we see chestnut, they grow like this, the tadpole. But in the forest, when you have no more chestnut trees, what is the replacement? This tree actually replace chestnut, maple tree, and or the beech tree, or maple tree. And if you give experiment here, the litter from maple or beech giving slower growth to the tadpole. So you can see that the tadpole actually like litter from chestnut. They don't really like maple or beech. So in this case, if you we have the chestnut tree gone, then probably the tadpole also might have problem. So in this case, if we test it with the GMO chestnut, actually the tadpole the same happy as the normal chestnut, and in fact they grow better than the maple or the beech one. So in this case, if you want to read it, this one, the life, death, and rebirth of a perfect tree. This is the story of American chestnut. Then the enzyme that we all use it in our daily, from the wheat for bread, for everything like this one. So chestnut poised for revival, and this we have to thanks to the transgenic work. Maybe we can save the chestnut later on. So here,
Excuse me.
turning the American. Okay, I will.
Professor Antonio. you have 2 minutes more to wrap up. Thank you, sir.
Okay, because I think I have a lead, right, from Pak Bambang Prasetya.
Yeah, so
I
you
think
have
I still have 5 more minutes. Okay, but I will make it as
Okay,
fast.
sir.
Yeah.
Okay.
Okay, because I got lead from previous speaker. Okay, that's okay. So turning the American chestnut into a GMO might be the only way to save it. And probably here, maybe just my slide. You can look at here how this slide looks beautiful here. But if you change the cows here with a tractor, maybe looks less beautiful. But that is the technology. And the farmer like to have that technology. But for us, we like this one, this because we think it is more beautiful. Even if you look at the house here, also, if this become a hotel with how many stories, maybe you will think this photo is not so beautiful. So that's why maybe we can put this air condition here in the hut and then make us better. So that is the technology. Sometimes have a romanticism, but not really in line with the real life of farmer. Okay, I think that's all. I give you this presentation and thank you for Dr. Evelyn for this. Okay.
Okay, sir. Thank you very much, Professor Antonios, for such a mind-boggling information that you've just shared with us. So we just have, for me to simplify matters, I just have to enjoy my yellow sweet corn and the spineless or the thornless durian. Yes, sir. So thank you very much for this information and updates on biotechnology and biodiversity conservation. Indeed, it's a very trending and never ending topic on biodiversity conservation, especially so that we do acknowledge biodiversity is life. And it's one of the major concerns worldwide, and it's one of the 17 Sustainable Development Goals. So thank you, sir,
Thank
for
you.
clearing
Okay.
some doubts on the GMO.
Thank you.
Thank you very much, sir. Okay, so our next speaker is Mr. Wadzanai Mandiveni. Sorry, I hope I pronounce it correctly. And he hails from Zimbabwe, South Africa. He is a member of the Secretariat for the Convention on Biological Diversity. Currently, he is the head of biosafety. Our third speaker has finished a bachelor's degree in biological sciences and a master's degree in Master of Science in molecular genetics, sorry. And sooner or later to finish his doctoral degree. His experiences and achievements include him being the biosafety regulator and biodiversity risk manager. He is currently working for the United Nations Environment Program as the head of the biosafety unit in the Secretariat for the Convention on Biological Diversity. Ladies and gentlemen, please help me welcome Mr. Wadzanai and his presentation on building capacity for effective participation in the Biosafety Clearinghouse of the Cartagena Protocol on Biosafety. Sir? Oh, sorry, ma'am. I'm really sorry. Please forgive me. Madam, what's the time? The time, next 30 minutes are all yours. Madam.
Great. Thank you very much, and good morning to you all. And thank you very much to the organizers for extending an invitation to the Secretariat for the Convention on Biological Diversity. Indeed, it's a great pleasure for us to be part of this very informative session. And before I begin, Madam Chair, I am so impressed by just the last two presentations. And I am wondering how I can get the distinguished professors to participate in global processes under the Cartagena Protocol on Biosafety. So thank you very much for extending an invitation to us, and good time of the day to you all. It's 11:38 p.m. here in Montreal. So you'll forgive me if I'm not able to stay for the whole session, but I will stay for this session. It's been very informative so far. So all protocol observed, and thank you once again. So the brief that we have is to provide an introduction to the Biosafety Protocol with a focus on the obligations relevant to the Biosafety Clearinghouse. And just before we proceed, just to give some background, at the United Nations Conference on Environment and Development, the big Rio Earth Summit that was held in 1992, the Convention on Biological Diversity was opened for signature. And this convention represents a global commitment to conservation of biological diversity, sustainable use of its components, and the fair and equitable sharing of benefits arising from the use of its genetic resources. What was also really critical to note is access to biotechnology and the sharing of its benefits. So the convention includes Article 16 on access to and transfer of technology, some of which is the technologies that we've already been introduced to this evening. And Article 19 speaks about the handling of biotechnology and the distribution of its benefits. And the Convention on Biological Diversity therefore forms the basis of international law on biosafety. The Biosafety Clearinghouse. So the Biosafety Clearinghouse was established by Article 20 of the Cartagena Protocol on Biosafety. And the Cartagena Protocol on Biosafety is a supplementary agreement to the Convention on Biological Diversity, and it represents the preeminent international instrument for regulation of biosafety issues around the world. The Biosafety Clearinghouse is the instrument that allows parties to exchange information on living modified organisms, and therefore is fundamental in facilitating the implementation of the Cartagena Protocol on Biosafety. At this point, I would just like to stress that in the context of the Cartagena Protocol on Biosafety, it being an environmental agreement, we use the term living modified organisms as opposed to genetically modified organisms. And this distinction is really meant to indicate the fact that we're talking about those organisms that are able to replicate in the environment. So we refer to living modified organisms and not genetically modified organisms in the legal instrument. But for all intents and purpose, and when you look at the national records and information shared on the BCH, you'll see the terms GMO and LMO are used interchangeably. So information sharing and the Biosafety Clearinghouse. So the Biosafety Clearinghouse was established to facilitate exchange of scientific, technical, and environmental and legal information on and experience with living modified organisms. And it also is used as a primary tool to assist parties to implement the protocol. The BCH provides access to information which is made available by the parties relevant to implementation of the protocol, as well as other international biosafety information exchange mechanisms. A visit to the Biosafety Clearinghouse will allow you to have access to some of the exciting research we heard from the previous speakers. Another important function of the BCH is that it requires each party, and we have at the moment 173 parties to the Cartagena Protocol. And so the Article 20 requires each of those 173 parties to make available to the BCH any information required under the protocol. In particular, you can find on the BCH laws, regulations, and guidelines for implementation of the protocol, bilateral, regional, and multilateral agreements, as well as, most importantly, summaries of risk assessments or environmental reviews that have been generated by the regulatory processes. You will also find final decisions that countries have taken with regard to importation or release of living modified organisms. The modalities of operation of the BCH, including all of its reports, activities that are considered and decided upon by the Conference of Parties, serving as the meeting of the parties, are kept under review. So if you would like to find out more about how the Biosafety Clearinghouse works, please visit the Biosafety Clearinghouse itself, but also have a look at Decision 1.3, which outlines the roles, characteristics, and administration of the BCH. It gives information on the role in providing access to information, the guiding principles to ensure inclusiveness and transparency, as well as the way that the Secretariat works in order to ensure that information is accessible, user-friendly, searchable, and understandable. And for those of you who are familiar with the BCH, you will be aware that in 2021, we launched a revamped BCH, which has greater functionality. And this is all the work that the Secretariat does to ensure that the BCH is functional. So the type of information that exists on the BCH, we have information in two broad categories. National records, which are published by the governments themselves, and they include information that is obligatory on parties, such as the national information relevant to the implementation of the protocol. Then we have other information which we call reference records, and this includes a number of biosafety-related resources and information which can be submitted by any registered user, and these are validated by the Secretariat prior to their publication. So the information that exists on the BCH, under the national records, you will find information on the national focal point, information on competent national authorities, information on the supplementary protocol on liability and redress competent authorities. You'll find biosafety laws, regulations, guidelines, and agreements, country decisions, or any other communications that countries may have, risk assessments generated through a regulatory process, national biosafety websites and databases. I think the next presentation after me will be showcasing one such national biosafety website, information on biosafety experts in the form of a roster of experts, as well as national reports on implementation of the protocol. All these national reports can only be published by BCH national focal points. And then we have the reference records. These are records that are independently published, and they include the Biosafety Virtual Library resources, where a number of publications on issues of relevance to biosafety are information on biosafety organizations, laboratories for detection and identification of living modified organisms, general information on living modified organisms themselves, information on genetic elements, organisms, risk assessments that have been generated by independent or non-regulatory processes, capacity building initiatives, as well as a newsletter. And this information can be submitted by any registered user, and I encourage participants to register to become users of the BCH and have access to this wealth of information, but also to share your information on the BCH. So this is what the page on the BCH looks like with the national records, and you will be able to simply go on. When you click, you are able to go inside. So the first obligation for national focal points and competent national authorities, this is information that is made available in terms of Article 19 of the protocol. This article requires each party to designate a national focal point who liaises with the Secretariat and provides official communication on behalf of the country. It also requires countries to designate one or more competent national authorities that are responsible for performing administrative functions required for the implementation of the protocol. And all this information is available on the BCH. Article 17 of the Protocol on Emergency Measures also requires parties to make available relevant details, setting out the point of contact, the purpose for receiving notifications, and who and how countries respond in this case. The biosafety laws, regulations, guidelines, and agreements that are made available. The Cartagena Protocol under Article 2 requires parties to take necessary and appropriate legal, administrative, and other measures to implement obligations under the protocol. Article 14 requires countries to make that where countries may enter into bilateral, regional, or multilateral agreements and arrangements for intentional transboundary movements of living modified organisms. And all of these agreements are also available and should be published on the BCH. And then, of course, Article 20 itself that provides an overarching obligation for countries to make this information available. Country decisions or any other communications, countries are obliged in terms of Article 20 to make decisions published on the BCH, in particular decisions on intentional introduction into the environment, decisions on living modified organisms for direct use as food, feed, or for processing, and any other decisions, declarations, notifications, or communications that countries may have under the Cartagena Protocol. This is just an example of what the page looks like and what decisions look like when you log into the BCH. Also available on the BCH is important information on risk assessments generated through regulatory processes. In terms of Article 20, parties are required to make risk assessments available. Risk assessments and environmental reviews of living modified organisms are done in accordance with Article 15 on risk assessment. And in this regard, risk assessments need to be done in a scientifically sound manner, taking into account recognized risk assessment techniques. Annex 3 of the protocol provides a general outline of principles, methodology, and the points which are necessary to consider when conducting a risk assessment. And again, all of this information is for all the risk assessments that have been undertaken are available on the BCH. We also have, in terms of Article 23 on public awareness and participation, an ability that countries may have national websites or databases that include publicly searchable internet sites on biosafety. Article 23 for public awareness requests parties to promote and facilitate public awareness, education, and participation. And the BCH is an important tool in this regard, and also to endeavor to ensure public awareness and education include access to information on living modified organisms. Another important component of the Cartagena Protocol is Article 33 on monitoring and reporting. And again, this article is implemented and the information is shared through the BCH. But under Article 33, each party shall monitor the implementation of its obligations under the protocol, and at intervals make these reports available. As you may know, we currently are have just completed the 4th reporting cycle, which was adopted by COPMOP 9 in 2018. And those national reports have been completed and the 4th assessment and review undertaken. We now are busy preparing for the 5th national report, which will be undertaken in 2026. But if you're looking for any information on national implementation, all of the reports from the interim report, the 1st, 2nd, 3rd, and 4th national report are all available on the BCH. And it's also possible using the analyzer on the BCH to compare progress in countries and check reports over time. So this is what the page looks like on the BCH, where you can access information. And this is how the national report template looks on the BCH. The next set of records that are on the BCH, again, are reference records. And as I indicated, these are the records that can be produced by any registered user on the BCH, so they don't need to come from national competent authorities or national focal points, and the Secretariat reviews these records before they are published. And these reference records are very good resources for not only exchange of information, for research purposes, and also for ensuring broader public participation. So we have reference records to facilitate exchange of scientific, technical, environmental, and legal information and experiences, and also to provide access to information made possible by parties. And the reference records available are the Biosafety Virtual Library resources, where journal articles and other important and pertinent papers are shared, information on biosafety organizations, the detection network, etc. So in conclusion, I really would like to once again recommend that you visit the BCH, which is at the core of the Cartagena Protocol on Biosafety and its implementation. And it's also the same tool that is used for monitoring the implementation of the Nagoya Kuala Lumpur Supplementary Protocol. And it's also the platform that we use for online meetings and online discussions and deliberations. We presently are having an online discussion which will start in a few weeks on gene-drive mosquitoes, which might be of interest to your partners. But I thank you for the opportunity to listen in and contribute to the discussions. Thank you, Madam Chair.
Thank you very much. Yes, Madam Wadsi. Oh, what's happening? So for this, for another enlightening and insightful presentation on how countries or institutions can be effective participants in the BCH, the Biosafety Clearinghouse of the Cartagena Protocol. So our fourth and the last but not the least speaker on the implementation of Biosafety Clearinghouse, the Indonesian experience, or in Indonesia, we have another lady expert, Madam or Ibu Elmi Kurniawati, SSI MPA. She hails from Tangerang Selatan, Indonesia. And currently she is the Senior Policy Analyst of the Directorate of Biodiversity Conservation for Species and Genetics at the Ministry of Environment and Forestry, Indonesia. She finished her bachelor's degree at the Faculty of Biology, Gadjah Mada University, and her master's degree from the Magister of Public Administration, Gadjah Mada University. So again, sooner or later, she will obtain her doctoral degree. Her experience and achievements include having participated or joined the Agriculture, Biotechnology, and Biosafety program of the Michigan State University in 2019, having joined the Asia Regional Biosafety Clearinghouse Training Workshop in New Delhi in 2020, and the training course on risk assessment of living modified organism for Southeast Asia and Pacific Asia in Manila, Philippines in 2022. Ladies and gentlemen, please help me welcome another lady expert on her topic on the implementation of Biosafety Clearinghouse in Indonesia. Ibu Ilmi, the next 30 minutes are all yours. Yes, Madam. Sorry, ma'am, you are muted. Please switch on your microphone. Or the organizing committee, can you please help? Madam Ilmi, Bu, your microphone is turned off. You are muted.
Oh, okay.
Thank you, Bu.
Thank you, Ms. Evelyn, for time for me to explain my presentation. Ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh. Good morning for all participants of the. I'm here. I'm sorry if my director, Ibu Mrs. Indra Exploitasia, can join the meeting, the workshop, because she has an urgent meeting with my ministry, and I am really pleased to her to explain the presentation. I have a presentation. Please share my presentation. Miss Evelyn, I'm sorry if I explain the presentation with mixed language.
It's
One
okay.
bahasa.
There
Yes.
is going to be an interpretation channel. Yeah.
Yes.
Also for the information of the other participants who are non-bahasa speaking, you can switch on the interpretation icon and you can choose English. Okay, Madam.
Yes. Thank you, Miss. Please share my PPT. Okay. I'm in the Secretary of Biosafety Commission, Indonesia Biotech Commission. We can explain the implementation of biosafety clearinghouse in Indonesia. This BGH in Indonesia by operate by Directorate of Biodiversity Conservation for Species and Genetics in Ministry of Environment and Forestry. Next. National regulation related to biosafety as law ratification of the Convention of Biodiversity, law number 21 of consent ratification of Cartagena Protocol on Biosafety, and law number 32 concerning environmental protection and management, law number 21 on animal, fish, and plant quarantine, government regulation concerning biosafety of genetically engineered products, President Decree of 29 of concerning the Biosafety Commission for genetically engineered products. This biosafety clearinghouse in Indonesia, based on Presidential Decree number 29 of concerning the Biosafety Commission for genetically engineered products. So regulation number 20 concerning the concerning amendment to Presidential Regulation concerning the Biosafety Commission for genetically engineered products. Next. Next. And the Indonesia Biosafety Commission for genetically engineered product is non-structural institution that's responsible to the president. This institution provide recommendation on biosafety of GHP to the authorized minister and or head of institution, and assist in carrying out supervision of the import and utilization of GHP as well as conduct an investigation of negative impact for or reported. So the minister is Minister Environment and Forestry, Minister of Agriculture, and head of Food and Drug Control Agency. And carrying out its duties, the institution is assisted by the biosafety technical team for GAP, the Secretariat of Indonesia Biosafety Commission for GHP, and the Biosafety Clearinghouse. So the Biosafety Clearinghouse is part of the Indonesia Biosafety Commission for genetically engineered product. Next. Please next. Yeah. The Indonesia Biosafety, okay. The Indonesia Biosafety Commission of genetically engineered product with decrees of the President of the Republic of Indonesia number 53 year 2022 regarding termination and appointment of membership of the Biosafety Commission of genetically engineered product. the head of the Indonesia Biodiversity Commission of Genetically Engineered Product is Professor Bambang Prasetya. You can the keynote speaker on this workshop. Okay. Next. This is the decision, President decision of Republic Indonesia. Next. It's bahasa. This the structure of Indonesia Biosafety Commission. Yes, Chairman of Indonesia Biosafety Commission for Genetic Engineering Product is Professor Dr. Insinyur Bambang Prasetya MSc. We have 3 vice chairman for food safety, vice chairman for feed safety, and vice chairman for environment safety. Vice chairman for food safety is in Food and Drug Control Agency. Vice chairman for feed safety in Ministry of Agriculture, and vice chairman for environment safety in Ministry of Environment and Forestry. Next. Okay. Other members of Indonesia Biosafety Commission for Genetically Engineering Product is Ministry of Marine and Fisheries, Ministry of Education, Culture, Research and Technology, Ministry of Trade, Ministry of Industry, Ministry of Health, and National Research and Innovation Agency is 3%. It's we can also call the green. So representative from university 3%, from Gajah Mada University, for Bandung Technology Institute, and Bogor Agriculture Institute. So next element of society is 4%. No, totally 19% in Indonesian Biosafety Commission of Genetic Engineering Product. Next. It's a technical team of Biosafety for Genetic Engineering Product. Next. Coordinator of technical team of Biosafety for Genetically Engineering Product, FH3. 1 is coordinator of technical team of food safety for in Food and Drug Control Agency. That's coordinator of technical team of feed safety for genetically engineering product is Ministry of Agriculture, and coordinator of technical team of environment safety for GHP in Ministry of Environment and Forestry. Next. Next. This the decision from decision letter from Professor Bambang Prasetya is with the member of technical team of Biosafety Commission for Genetically Engineering Product. It's food safety, in the feed safety, and the environment safety. In environment safety, we can have 2, 3 and space. This is plant and fish. It's 2 and animal. So and microbiology, microbe. Next. Secretariat of Indonesia Biosafety Commission for Genetic Engineering Product is in Ministry of Environment and Forestry. Next. This since the Secretariat take place of the Directorate of Biodiversity Conservation for Species and Genetic, Ministry of Environment and Forestry, and lead by my director as Mr. Indra Exploitasia. So to assess implementation of Indonesia Biosafety Commission test, support administrative matters and operationalization of Biosafety Clearinghouse. So we in Secretariat day to day to operate Biosafety Clearinghouse. Next. So in Indonesia Biosafety Clearinghouse can be accessed through Indonesia BGH and LHK Go ID. So can everyone to can be access this the web. Okay. Next. Clearinghouse, and we have 3 clearinghouse in Indonesia. One is a clearinghouse mechanism based on convention biodiversity convention. So we have biosafety clearinghouse based on Cartagena protocol, and we have access benefit sharing clearinghouse based on Nagoya protocol. So Indonesia have 3 clearinghouse into implementation convention of biodiversity. Next. The Indonesia Biosafety Clearinghouse, we call BGH, is part of the Indonesia Biosafety Commission for genetically engineering product in managing and presenting information to the public. Indonesia BGH, managed by Directorate of Biodiversity Conservation of Species and Genetics, Ministry of Environment and Forestry, which can be accessed through www.indonesia BGH LHK Go ID. Next. It's a working group on Indonesia clearinghouse mechanism on this decision on the process for renewal. That is the steering committee, technical team, secretariat, this implementation group for Biodiversity Convention, for Cartagena Protocol, Nagoya Protocol, and governance. It's the decision letter is on the process for renewal. Next. This implementation group on Cartagena protocol is the personnel is head of Directorate of Biology Conservation, Secretariat. So one Food and Drug Control and Green and Ministry of Agriculture, so and so on. The task in a group of Cartagena Protocol is develop information network related to biosafety between biotechnology developers and the public. It's food expert network delivering the result of risk assessment, analyzing community input before awarding certificate, updating information data for genetically engineering product, develop monitoring network and evaluation post-release monitoring, assess NFPs on formulating national report, the implementation of Cartagena protocol. So this team is on the process for renewal too. Next. The implementation of the Indonesia BGH published a summary of risk assessment for genetic engineering product to the public for 60 days to get input before the plenary session of the Indonesia Biosafety Commission for genetic engineering product. So we can upload the summary of risk assessment. Can somebody can be input for the web to information result or summary of this assessment. Indonesia BGH have contained the latest biotechnology development, including genetic engineering product in Indonesia, activity and news related to the biosafety issues in Indonesia. Indonesia BGH published biosafety recommendation for GMO and for genetic engineering product that have been issued by the authorized minister on the head of institution. Indonesia BGH is connected directly to the BGH central portal and provide information and mandated by Cartagena Protocol. Next. Now, this guidance page, there's panduan is contain information about regulation on biosafety. You can find the regulation related on biosafety. You can solve this panduan. Yes, next. So in the BGH, in the BHK, we can publish news about the biosafety plenary session for genetically modified product. We can plenary session, maybe we can risk assessment, we can risk management and so on. We can publish in the BGH. Next. It is important, but BGH can present a summary of biosafety assessment result. So we can get information about biosafety assessment. This public can give information or give complaint and so on this product before plenary session of Indonesia Biosafety Commission. Next. Next, the public can provide feedback through public participation during the specified on and close date of the summary of the safety assessment result. So, public can give feedback on the web, so we can read the feedback and plenary session of Indonesian Biosafety Commission for genetic engineering product. Next. This decision letter contain data of genetically modified organism or genetically engineering product from the party that have submitted on application for environmental food feed safety assessment of GMO. So we can find the decision letter for the BGH. Next. Next. This in BGH can direct link to the BGH central portal under Cartagena Protocol. So we can have in the Indonesia based in biosafety clearinghouse, we can click to national database, so we can have open the link to the BGH central portal under Cartagena Protocol. So we have some information the genetic GMO or the genetically engineered product. Okay, Miss Evelyn, thank you. I hope my presentation can clear this implementation for Indonesia Biosafety Clearinghouse. Thank you.
Thank you very much, Madam Ilmi, for the information on the remarkable efforts of the government of Indonesia in the implementation of biotechnology. So, ladies and gentlemen, we have just been enlightened and updated on the important roles of biotechnology in food security and biodiversity conservation. We were also made to understand on how we can effectively participate as countries or institutions in the implementation of the Cartagena Protocol on Biosafety. And also, we were also presented and enlightened on the Indonesian experience in the implementation of this biosafety protocol. So now We would like to request our resource speakers, our seasoned resource speakers, to open their videos, and we encourage the participants to air out their questions, their queries on our topic on biodiversity, biotechnology. Okay, thank you very much, distinguished resource speakers, for sharing those valuable information with us. So now we proceed with the Q&A, the question and answer. So please understand that we have the YouTube live, and we have the on-site participants and the online participants. So I would like to request the organizing committee to please post on the screen the questions. Our resource speakers can answer, can give their opinions, suggestions on the question, any one of them, if the question is not particularly directed to a particular speaker. But even then, if the question is directed to a speaker, the other speakers can still freely join in the discussion or in answering the queries. Thank you very much, sirs and madams. Okay, so the first question, shall I read, is from Dr. Supriyanto of Biotrop, right? Yes. So this is directed to Professor Antonios. Sir, thank you for your presentation. Do you think that the human being could consume all DNA material due to the genetic, genetic, right? Similarity between human and other living? Yes, sir. Professor Antonios.
Okay, I would like to answer that one, but unfortunately I'm on the road. But it's okay. I actually already wrote the answer to Pak Supriyanto. Basically, for DNA, it's no concern because we eat DNA every day. When you eat any food, that's always contain DNA. It's almost impossible. You can think there is food without DNA. Probably Coca-Cola has no DNA, but water like that. But if you eat something, you always have DNA in it. So eating DNA is no concern at all. So we use that one every day. But the one that you need to be of concern is the protein, the product. Whether this one maybe might have something like whether toxin or maybe allergen or something. But DNA is completely nothing to worry about. Okay, thank you.
Thank you very much, Professor Antonios. Pa or Dr. Supriyanto, are you satisfied with the answer, sir? So there is another question from Dr. Supriyanto, and this is directed to Madam Nayati or Madam Wadsi. How do you harmonize the term between LMO and GMO in the operational level? Madam?
Thank you very much. And I had responded in the chat, but I will speak again. And I think that in everyday use and for everyday interpretation, the two are used synonymously. So LMOs, GMOs doesn't make a difference in everyday use and in interpretation. However, in interpreting the protocol, it becomes important because the L of living means that it needs to be something that has the capability to transfer its genes from one one organism to another. So it needs to be able to be a biological entity capable of transferring or replicating genetic material. And this is where in the protocol the issue of scope is important. So that's how we reconcile it in everyday language. They're used synonymously, but when it's time to interpret legally, then it is important to make the difference. And so you will find in all of our legal, in all our decisions and in all of our processes, we will refer to living modified organisms and not to genetically modified organisms.
Thank you very much, Madam Wadsi. Actually, if you are the one who raised the question and you want to have a further question, you are free to speak. So just open your microphone. You are recognized if you want to have a follow-up question on your question. So let's now proceed to the next question. This is directed to Professor Bambang Prasetya from another Professor Bambang Purwantara. Professor Bambang Purwantara, would you like to raise your own question, sir? Or do I have to read? Sir?
Well, thank you. I think I write down well that Bambang, following our discussion during the focus group discussion yesterday, held by the National Food Agency Bapangan in Jakarta, the Deputy Director of Bapangan raised the concern that speeding up the process of evaluation of GM product is something must to be speed up to facilitate the industry with more friendly processes. Do you have any recipe to reduce time spent, which is in some way is very long, and make the industry are rather feeling painful to head the situation? Please, Pak Bambang.
Thank you.
Thank
you, Prof. Bambang, for the question. I think it is very important question rising in the focus group discussion yesterday in National Agency for Food. My answer to the first, second answer, to answer. Firstly, is maybe related with the regulation. It is our regulation is quite long. Yeah, we need to revise. And I know from the Secretariat will prepare the revision of the regulation of the PP 21. But it is still not enough time, but I think because the regulation changing is take time. The second way we will speed up in the our business process based on the existing rule to make quite faster. But sometimes this problem is the data, the availability data from the proponent is not so enough. Therefore, sometimes take time to fulfill the needed data. Therefore, I recommend it to the proponent or the researcher that I already invite to make more proposal. That's better communication to provide a data for the assessment. There is Bu Elmiati also here know well about this situation of the communication and the data availabilities of from the proponent. I think this is my answer, Pak Bambang. Thank you.
Yes, thanks, Pak.
Thank you.
Okay, thank you. Pak Bambang Prasetya, maybe, yeah, there's the need to reduce or shorten the bureaucracy.
Yes.
Okay, so next question is again directed to from Professor Bambang and directed to Professor Antonios. Sir, it's interesting to know that chestnut has also their problems with blight fungus and already use biotechnology approach through 0x0XO gene to combat the problem. Is that any important state crop trees which have big economic impact such as oil palm, coffee, cocoa, or cacao, which suffer from any disease which need any biotechnology approach? Sir, Professor Antonio?
Yes. Okay. I think that is very good point that and exactly what I want to point out for the example for the chestnut that can be used for any kind of other trees that have problem, like for example oil palm and also cacao, everything related with that. But then we need a research related with that. For example, like transformation system, and then how to introduce the gene like that. But if we did not initiate this one, then we might just wait. Then it takes a long time. It is very important to anticipate like this. For example, like that chestnut. Actually, that one will help to preserve something that is already extinct. Maybe if there is no way, then they become extinct, or they just replace with the Chinese chestnut. So if they wanted only the American chestnut, there is no way, because the fungus is already ravaged and then killing all of the trees like that. So this is a good thing that we have to facilitate many research and also some approach related with the genetic engineering for conservation. Okay, thank you, Dr. Evelyn.
Thank you, sir. So the next question is directed to Dr. Wadsi. Again from Professor Bambang. Biosafety clearinghouse is very important element of biosafety assessment of the GM project, or product, or genetically modified product. Ideally, as you mentioned earlier, BCH can be as a portal to show the access every party, including industry, to understand the process of GM product evaluation. It is also functioning as risk communication portal. According to our experience, the public participations to give comment on any GM product on the pipeline for approval are very low. Would you please share any successful BCH in some countries with which are quite active on public participation? Madam Wadsi?
Thank you, and thank you for this very important question. We have seen that this is a concern for many countries, and working together with the Aarhus Convention on Public Participation, we've prepared a pocket guide to assist countries with public participation and giving them some tools that they can use to enhance public participation. So I recommend the pocket guide as a tool for the countries to make use of, and also to look at some examples of good public participation with the Aarhus countries. And one of the countries where I have seen good input, an example to look at would be Norway. It has a good system for public participation, and is a good example that you could look at if you wanted to see how that works. One of the big challenges which countries raised in the fourth national reports and the assessment and review, and even in previous years, is the resources required for public participation and the need for countries to first educate the public on what is going on before the public is empowered to actually participate in the decision making. So at the end of this year, we will be having a meeting in Geneva together with the Aarhus Convention to train countries on how to enhance public participation in decision making for living modified organisms. So this is a concern, and it remains a concern, and it's an area of growth for all of us. Thank you.
Thank you very much, Madam Wadsi, and please keep us posted on that event. So, yes, and the next question is from a senior high school student, Ms. Apriyansha from Bujung Pichung Chanjur Senior High School. Ms. Apriyansha, would you like to raise your question yourself to Dr. Professor Antonios? Okay, I will do it myself. Professor Antonios, there's a question here from a senior high school student who would like to get involved. So her question is that, what is the role, her role, or the role of the students so that biodiversity is maintained? Sir?
Okay. Okay. I think for the role, the question is like for the role, I think there can be a leader, okay, like the role. But for the conservation, this one will be so many things involved, so many perspective, everything like that, including land use, the change of land use or something. But for senior high school, I think better that you look for the traditional and ethnic food or practices. So like we should try to look at what is with the wisdom can be obtained from the traditional or ethnic application, and we try to look at the good thing of that. For example, if you are a senior high school student, we know that the food usually in Indonesia usually wrap or using banana leaf as a become more expensive because becoming more and more rare because people export it or they use it in the city like that. So maybe we have to preserve the way we use it, but not using like plastic. For example, if you have some lemper wrapped with banana leaves, try to use always banana leaves or something use bamboo leaves. We have to use it. So then we can force that the bamboo or the banana that type will be planted to conserve with your own role as a senior high school, right? Or because still in senior high school. Of course, if you go to the later on become a leader, then you can have more things to do with the conservation of biodiversity. But first, maybe you should look at the what we have already in our local wisdom. The local wisdom has a lot to do with the conservation of biodiversity because they use everything that is related with sustainability. I think we have to pay attention on the like this traditional or local wisdom to look at how we can use it for preserving biodiversity. Thank you.
Thank you very much, Professor Antonio. Is there anyone else among our resource speakers who would like to answer or give her or his opinion about the question of our high school student here?
Perhaps I can just say something and really to commend the young person. In December last year, we adopted the new global agreement, the Kunming Montreal Global Biodiversity Framework, with 23 action goals for the next 8 years. And one of the things that this agreement has, which it didn't have before, was the concept of intergenerational equity. And we had young people playing a key role in making demands of nature and making demands of policymakers to take into account the views of young people. So I think that as a young person, you can certainly continue to advocate, to empower yourself and to speak out and take biodiversity conservation matters to heart. The world is seeing unprecedented biodiversity loss, and making the case for biodiversity and just raising awareness amongst your peers to make and take better choices is something that is really to be encouraged. So I commend you for having even the question, and for you to keep asking the question and holding others accountable. Thank you.
Yes, anyone else? Professor Bambang, you want to add something to the query of our
I think it's enough. I think. Thank you.
Okay. Okay, so let me voice also my opinion regarding that question, since I am a teacher also and I advocate biodiversity conservation among my students. So I would like to say to Ms. Apriyansha that as a student, you really have a role to play. As the saying goes, we think globally, and that's biodiversity conservation. It's a global issue, and we act locally. So as an individual, we have influence among our peers, among your classmates, among your friends, and actually you can start with, as an individual, you can start with information campaign on the value of biodiversity. Why is biodiversity important? So by then, if your peers, your friends understand what is the importance of biodiversity, then they may be encouraged to also participate in the conservation of our biodiversity. Okay, so Dr. Romy, I've seen no more questions posted on the screen. So
there are 2 questions I'm still putting in the.
Oh yeah, where is it? Sorry, I cannot see it. There are. Oh, this one directed to Professor Antonios from Prima Gandhi, Tokyo, Japan. Is it?
Yes.
Yes.
Is it the one, Romy? So the question is, does the use of GMOs potentially eliminate local plant varieties, or in other words, the loss of biodiversity? Yes, Professor.
Okay. This question actually, the answer can be yes and or no, depend on how we do it. Even without GMO, even without biotechnology, our monoculture, like you cut the forest to make rice field, or to make oil palm plantation, for example. That one, no biotechnology, no GMO. If you did not manage it correctly, of course, this one will reduce so much diversity there. But if we can manage correctly, even though this one GMO or non-GMO, we can actually do it and even preserve it. Like, for example, I told you about the chestnut case. the chestnut is almost extinct. But probably with the help of DNA technology, we can restore the forest in the northeastern part of the United States, for example, in that case. And this can give us also some insight on how we do it, the other things. So depends on how we manage. So that's why it is important. We have to consider how we evaluate things before we embark on that type of implementation. Okay, thank you.
Okay,
Doctor.
thank you, Professor Antonio. So the last question here on the screen is directed to Professor Bambang from Dr. Supriyanto. I did some research on sorghum for adaptation to climate change, especially for drought tolerance. How to register those research results? Professor Bambang?
Yes, thank you to Dr. Supriyanto. Related the new variety from sorghum, when you already decide of the research and you have already a new variety drought resistant, when it is belong to GMO, you can apply to the committee or through the secretariat. There are certain forms you can do for that because you can consultation. But when it is belong to the gene expression, also you can also apply to select whether it is GMO or not. But what is important is the availability data, the needed data. You must be provided for to send the secretariat. It is very important because sometimes it take long because the data is not enough to assess. I think this is the answer for me related to the registration of this research. Thank you.
Okay, so Pak Romi, there's no more questions. So
Yeah. Yes, ma'am, actually in the chat box. But may I have a question? May I give a question actually? I
yes,
tried to
please.
raise my hand in the Zoom, but I don't know how to raise. I don't know where is the tools actually. May I
Okay,
have
go ahead,
questions?
Dr.
Give
Romi.
questions. So yeah. So actually, this is I have a question. This is actually the general questions about the biotechnology, about the advance or the advanced update, the update of the biotechnology. By questions actually that I have said before the first activity that I said, the technology of biotech or the biotechnology, it's actually rapid. It's really rapid. The advance is really advanced, rapid. So it's developed really rapid. The question is because like we know exactly actually 10 years ago in 2013 we know like the Jennifer Doudna, Emmanuel Carpentier has already found the CRISPR-Cas9, and then they also the genome editing technology, and then it's starting used for some research and starting used for the sums. How I remember in Arabidopsis there are several research used then in the populus, and I think I don't know how, but in the food products and in the products right now. The question is, I always wondering, and it's myself, is CRISPR-Cas9 is actually considered as the GMOs, or is it not really considered, or is it already be regulated in not only national, or maybe it's already regulated in internationally or something like that? Maybe that's maybe the questions for me. Thank you very much.
Thank you, Pak Romi. So who would like to
Okay.
answer?
Okay. Maybe I would like to give some answer for that one. Because this is also very interesting. You can see in the US they have engineering for apple. So the apple, when you cut, that will not become browning. So because browning, they don't like it when you slice it. This is using genetic engineering, the conventional one. So when they go for regulation, they enter the market after 5 years. So after 5 years, the apple enter the market. The same thing happened to a mushroom. This is a mushroom, the button mushroom. The button mushroom usually white, but if you become brown, people don't like it, the consumer. So then people actually make genome editing for this one. The same one similar to the one in apple. This is only change one nucleotide. What happened? Those one actually already in the market without people realizing this, because you cannot differentiate between what is original then the spontaneous mutation. So this is just like natural like that. So they are already in the market. No, like escape, escape from regulation. This is the button mushroom for the same thing. So yes, so finally the genome editing will become a tool that makes us more and more understand the to avoid the genetic engineering that is more cumbersome or something. So this one will be a good way to avoid maybe long relation or something. But still, this one actually from the standpoint, you change it one nucleotide. Okay, so I think I give only this one as an example. But for CRISPR, you see, not just for animal and plant. You see, in successful to create a human baby, now it's already child, maybe 5 years old, that is free from HIV by genome editing. Of course, the researcher has been protested by all over the world, but that is already done. You can check it that they use genome editing to edit the human genome, not just plant or animal. Okay, thank you.
Thank you very much, Professor Antonio. So one last question. Everybody satisfied, clarified? Okay.
Yep.
So finally, is there a question, Pak Romi?
Yeah, actually, I'd say about about this CRISPR-Cas9, about this genome editing. Maybe I also the next question is like, is the regulation is actually follows the development of technology from the biotech, specifically from the biotechnology, because the development of biotechnology seems very rapid, seems very quick. How I know CRISPR-Cas9 2013. Now I know in 2020 or 2021 there's already something somehow like similar like CRISPR-Cas9. I think it's called Omega. This kind of the genetic engineering is really rapid developing. The question is the regulations is really follows the technology, or it just lets the technology fall, lets the technology develop and the regulation just follow later. That's maybe the questions from me.
Any comment on that, Professor?
Yeah, thank you for Romi Ardensyah. I think the regulation actually followed by development of the science and technology, including biotechnology. But sometimes after we set up the regulation, so the research can also follow the regulation, so that they can spare the time to spend, not wasting time, because it is for example, the genome editing is the good case. Yeah, the genome editing is already exist, and then regulation follow the new trend of the biotechnology in terms of genome editing. After that, when the regulation already there, please researcher activity, please make a lot of research as needed from the society, I think. Thank you.
Okay, so finally, thank you everyone for your active participations. Professor Bambang, Professor Antonios, Madam Wadsi, Madam Ilmi, thank you very much for the valuable information that you have shared with us, the participants, and everybody in this Zoom. Indeed, this kind of information disseminations are one of the necessary steps for Biotrope to achieve its vision to become a reputable reputable center in enriching and promoting the real values of tropical biology in Southeast Asia and beyond. So without any more delay, I would like to thank you very much for everyone's active participation. Thank you, organizing committee, for making this event happen. And for everyone involved, cheers. And more power. Good job. So I would like to turn over the floor to Miss Alia, our MC, our lovely MC. Over to you, Miss Alia.
Thank you, Evelyn.
Where's Miss Alia? Or do I have to turn it over to Dr. Romy?
Okay, thank you, Bu Evelyn, Miss Evelyn. Thank you for leading our discussion so comprehensive for this session. So really interesting with our moderator and our honorable speakers, of course, there Professor Dr. Bambang, Professor Antonius, and speakers from head of Biosafety Secretariat Convention, etc. So thank you, ladies and gentlemen, for joining our session. And once again, we would like to say thank you for our moderator, Bu Evelyn. Thank you very much. And give applause maybe for Dr. Evelyn for leading this discussion very well. So ladies and gentlemen, we would like to move for our session that will be lunch break, and we will start for the next session at 2 p.m. and for all the participants can join this session for 2 p.m. And before that, we have the certificate for our speakers and moderators. So please present the certificate. Okay. Certificate from Dr. Zulhan Shah. For Dr. Alfred, our moderator today. The certificate for Professor Bambang Prasetya. And this is for Professor Antonius. So this is for Wazanaim. Okay, thank you. So that is our certificate for our speakers and moderators. So we will meet at 2 p.m. For all the participants can join our next session at 2 p.m. And we will have a lunch break first. And our moderator next session that will be Professor Bambang Purwantara. And thank you very much. And have a lunch break. So I'm Alia and we will meet at 2 p.m. Good afternoon. Wassalamualaikum warahmatullahi wabarakatuh. Thank you, Bu Evelyn.
Thank you, Ibu Evelyn.
You're welcome, Pak Romi, Miss
Thank
Alia.
you, Bu Evelyn. Thank
very
you.
much.
Thank you very much. Thank you, Bu Evelyn. Thank you for the speakers for the participants also.
Good job,
We will meet again.
everyone.
Yeah, we will meet again. We have still one session after this. Thank you.
Takdir cinta yang ku pilih, meskipun tak mungkin, walaupun ku mau membawa kamu. Oh, jika ku bisa, ku akan kembali, ku akan berubah. Takdir cinta yang ku pilih, meskipun tak mungkin, walaupun ku mau bawa kamu lewat mesin waktu. Kan ku bawa wajahmu, kan ku bawa namamu. Ku ingin tidur dan bermimpi malam ini, di sini di kamar ini, sendiri melintas sepi. Gosok masang rambut dan gaun malam, ku tak peduli. Di sana kau berdua, di sini aku yang sendiri. Di sana kau tersenyum, di sini aku yang menangis. Jangankan untuk bertemu, memandang pun saja sudah tak boleh, apalagi menyanyi bersama bagai hari lalu. Jangankan mengirim surat, menitip salam pun sudah tak boleh. Ternyata memang kau tercipta bukan untukku. Oh, kan ku simpan wajahmu, kan ku ukir namamu, kan ku buktikan kesetiaanku padamu. Biarlah di sini sendiri merajut hari-hari. Bukankah esok atau lusa mati pun aku sendiri. Jangankan untuk bertemu, memandang pun saja sudah tak boleh, apalagi menyanyi bersama bagai hari lalu. Jangankan mengirim surat, menitip salam pun sudah tak boleh. Ternyata memang kau tercipta bukan untukku. Oh, kan ku simpan wajahmu, kan ku ukir namamu. Jika saat nanti kau telah hidup sendiri dan dunia ternyata tak seperti harapanmu, ku ada di sini menjadi rumah yang selalu menanti kepulanganmu. Kelak kau akan jadi orang tua seperti aku, yang ingin anakmu bahagia dengan hidupnya. Bila bentakan kecilku patahkan hatimu, lebih keras dari itu dunia akan menghakimimu. Ku bentuk dirimu menjadi engkau hari ini. Kau harus kuat, kau harus hebat, permata hatiku. Nah, kan tiba waktu kau harus tentukan jalanmu, yang mungkin tak searah dan indah di mataku. Apabila terjadi, berjanjilah kau akan selalu menjadi dirimu sendiri. Kelak kau akan jadi orang tua seperti aku, yang ingin anakmu berkuasa atas hidupnya. Bila bentakan kecilku patahkan hatimu, lebih keras dari itu dunia akan menghakimimu. Ku bentuk dirimu menjadi engkau hari ini. Kau harus kuat, kau harus hebat, permata hatiku. Bila bentakan kecilku patahkan hatimu, lebih keras dari itu dunia akan menghakimimu. Ku bentuk dirimu menjadi engkau hari ini. Kau harus kuat, kau harus hebat. Aku adalah jemari dan ibumu penanya, dan kaulah puisi terindah yang pernah tercipta. Semoga belayan kasihku lembutkan hatimu. Kau harus megah, kau harus indah, kau harus kuat, kau harus hebat, permata hatiku. Jangan pergi dari diriku, tak sanggup harus hidup tanpamu. seperti yang terjadi kemarin. Kini harus aku lewati langkah demi langkah yang menyakiti, membalut luka lagi yang kau tahan hingga kini. Habis sudah nafasku menyebut namamu. Takkan hilang cintaku padamu, takkan hilang walau kau memilih pergi, takkan hilang sampai di ujung waktu. Cintamu tak terganti, takkan hilang cintaku padamu, padamu. Takkan hilang cintaku padamu, takkan hilang walau kau memilih pergi, takkan hilang sampai di ujung waktu. Takkan hilang cintaku padamu, takkan hilang walau kau memilih pergi, takkan hilang sampai di ujung waktu. Pernah ku sangka ini terjadi, kisah cinta yang suci ini kau tinggalkan begitu saja. Sekian lamanya kita berdua, ku sangka begitu cepat berlalu. Untuk mencari kesombongan diri, lepas segala yang pernah kau ucapkan, kau tinggalkan aku. Pergilah kasih, kejarlah keinginanmu. Selagi masih ada waktu, jangan hiraukan diriku. Aku rela berpisah demi untuk dirimu. Semoga tercapai segala keinginanmu. Ku sangka begitu cepat berlalu untuk mencari kesombongan diri, lupa segala yang pernah kau ucapkan. Kau tinggalkan aku, pergilah kasih, kejarlah keinginanmu. Selagi masih ada waktu, jangan hiraukan diriku. Aku rela berpisah demi untuk dirimu. Semoga tercapai segala keinginanmu. Pergilah kasih, kejarlah keinginanmu.
Naka-break siguro ini.
Selagi masih ada waktu, jangan hiraukan diriku. Aku rela berpisah demi untuk dirimu. Semoga tercapai segala keinginanmu. Kejarlah keinginanmu. Selagi masih ada waktu, jangan hiraukan diriku. Aku rela berpisah demi untuk dirimu. Semoga tercapai segala keinginanmu. Berdiri memutar waktu, teringat kamu yang dulu ada di sampingku setiap hari. Jadi sandaran ternyaman saat ku lemah, saat ku lelah. Tersadar tinggal sendiri, merenungi semua yang tak mungkin. Saat putar kembali seperti dulu, ku bahagia tapi semuanya hilang tanpa sebab kau hentikan.
Luway-luwayon mo.
Pun sama seperti aku, tak ingin cinta usai di sini, tapi mungkin inilah jalannya.
Internet connection doon. Ikaw internet dahil. Ano naman?
Terluka, menangis, tapi ku terima semua keputusan yang telah kau buat. Satu yang harus kau tahu, menanti kau tuh kembali. Tinggalkan semua di sini.
Nagbayad ba 99 pesos, ano?
Ku tahu kau pun sama seperti aku, tak ingin cinta usai di sini, tapi mungkin inilah jalannya, harus berpikir.
Ano man yung sa ano ko, ano, screenshot? Screenshot? Ano ito? Bigyan na kayo ng ano. Hindi ka.
Kau ucapkan engkau tinggalkan aku. Kadang hati berharap ku terbangun dari sebuah mimpi. Ini bukan kenyataan, namun adanya meninggalkanku karena kesalahanku. Mungkin ingin bertemu masih ada, ingin memeluk masih ada. Sayang kini tak bisa, kau telah memilihnya. Mungkin saat hatiku masih sayang, setelah ku memutus cinta, dan kini ku menyesal. Rindu hanya di dalam hati. Andaikan aku tak tergesa memutuskanmu karena egoku, hati ini tak akan begini. Setiap ingat rasanya ingin kembali. Mungkin ingin bertemu masih ada, ingin memeluk masih ada. Sayang kini tak bisa, kau telah memilihnya. Oh, mungkin saat hatiku masih sayang, setelah ku memutus cinta, dan kini ku menyesal. Rindu hanya di dalam hati. Ingin memutar waktu, ingin mengulang lagi. Aku tak bisa berbalik lagi. Begitu juga Salah ku memutus cinta, dan kini ku menyesal. Rindu hanya di dalam hati. Rindu hanya di dalam hati. Di hati kau tetap indah. Selalu di hati. Selalu indah dilupakan, selalu sedih dikenang. Setelah aku jauh berjalan, betapa hatiku bersedih, mengenang kasih dan sayang. Setulus pesanmu kepadaku, andaikan kau datang kembali, jawaban apa yang kan ku beri? Adakah jalan yang kau temui untuk kita kembali lagi? Bersinarlah bulan purnama, seindah serta tulus cintanya. Bersinarlah terus sampai nanti lagu ini ku akhiri. Betapa hatiku bersedih, kasih dan sayang. Setulus pesanmu kepadaku, andaikan kau datang kembali, jawaban apa yang kan ku beri? Adakah jalan yang kau temui untuk kita kembali lagi? Bersinarlah bulan purnama, seindah serta tulus cintanya. Bersinarlah terus sampai nanti lagu ini ku akhiri. Kau datang kembali, jawaban apa yang kan ku beri? Bersinarlah bulan purnama
Kacen.
seindah mata tulus yang tak bersinarlah terus sampai nanti lagu ini akhiri. Waktu pertama kali melihat dirimu hadir dan hati ini inginkan dirimu, hati tenang mendengar suara indah menyapa, telaranya hati ini aku sangka. Rasa ini tak tertahan, hati ini selalu untukmu. Terimalah lagu ini dari orang biasa. Cintaku padamu luar biasa. Aku tak punya bunga, aku tak punya harta. Yang ku punya hanyalah hati yang setia, tulus padamu. Hari-hari berganti, kini cintaku melihatmu, memandangmu bagai bidadari. Rasa ini tak tertahan, hati ini selalu untukmu. Terimalah dari orang biasa, tapi cintaku padamu luar biasa. Aku tak punya bunga, aku tak punya harta. Aku punya hanyalah hati yang setia, tulus padamu. Terimalah lagu ini dari orang biasa. Terimalah lagu ini dari orang biasa. Tapi cintaku padamu luar biasa. Aku tak punya bunga, aku tak punya harta. Yang ku punya hanyalah hati yang setia. Yang ku punya hanyalah hati yang setia. Seluruh jiwa telah berserahkan. Kami janji setiaku jangan jadikan semua ini alasan kau menyakitiku. Meski cintamu tak hanya untukku, tapi cobalah sejenak mengerti. Bila rasaku ini rasamu, sanggupkah engkau menahan sakitnya terhianati cinta yang kau jaga? Coba bayangkan kembali betapa hancurnya hati ini. Kasih semua telah terjadi. Memohon jangan jadikan semua ini alasan kau menyakitiku. Meskipun cintamu tak hanya untukku, aku coba sejenak mengerti. Oh, bila rasaku ini rasamu, sanggupkah engkau menahan sakitnya terhianati cinta yang kau jaga? Coba bayangkan kembali hancurnya hati ini. Kasih semua telah terjadi. Bila rasaku ini rasamu, sanggupkah engkau menahan sakitnya terhianati cinta yang kau jaga? Coba bayangkan kembali betapa hancurnya hati ini. Kasih semua telah terjadi. Semua telah terjadi. suasana baru. Kau mampu tenangkan aku di saat risau dalam hatiku. Lembutnya sikapmu menenangkan hati ini. Terbuai aku terlena oleh dirimu, oleh diriku. Jantungku bergetar saat engkau ada di dekatku. Mungkinkah diriku telah jatuh cinta pada dirimu? Sebisa diriku mencoba untuk melupakanmu, namun ku tak bisa. Kau pun selalu ada dalam hatiku. Dan biarkan semua mengalir apa adanya. Ku yakin kau pun pahami perasaanku, oh, jantungku bergetar saat engkau ada di dekatku. Mungkinkah diriku telah jatuh cinta pada dirimu? Sebisa diriku mencoba untuk melupakanmu, namun ku tak bisa. Kau pun selalu ada dalam hatiku. Jantung pun bergetar saat engkau ada di dekatku. Mungkinkah diriku telah jatuh cinta pada dirimu? Sebisa diriku mencoba untuk melupakanmu, namun ku tak bisa. Kau pun selalu ada dalam hatiku. Hadirlah dirimu, berikan suasana baru. Ku hampiri jalan yang kita lewati setiap hari kita di sini. Ku menanti hadirmu untuk kembali, hanya kenangan yang tersisa di sini. Namun sekarang kau telah pergi dan ku yakini kau takkan kembali. Mungkin hari ini, hari esok atau nanti, berjuta memori yang terpatri dalam hati ini. Mungkin hari ini, hari esok atau nanti, tak lagi saling menyapa, meski ku masih harapkanmu. Kau telah pergi dan ku yakini kau takkan kembali. Mungkin hari ini, hari esok atau nanti, berjuta memori yang terpatri dalam hati ini. Mungkin hari ini, hari esok atau nanti, tak lagi saling menyapa, meski ku masih harapkanmu. Sesungguhnya hatiku tak sanggup menerima dan lupakan segalanya. Mungkin hari ini, hari esok atau nanti, berjuta memori yang terpatri dalam hati ini. Mungkin hari ini, hari esok atau nanti, tak lagi saling menyapa, meski ku masih harapkanmu. Meski ku masih harapkanmu. Ku relakanmu. Saat ku sendiri melihat foto dan video bersamamu yang telah lama ku simpan. Hancur hati ini melihat semua gambar diri yang tak bisa ulang kembali. Ku ingin saat ini engkau ada di sini, tertawa bersama seperti dulu lagi. Walau hanya sebentar, Tuhan tolong kabulkanlah. Bukannya diri ini tak terima kenyataan, hati ini hanya rindu. Segala cara telah ku coba agar aku bisa tanpa dirimu, namun semua berbeda, sulit untuk menghapuskan bersamamu. Ku ingin saat ini engkau ada di sini, tertawa bersamaku seperti dulu lagi. Walau hanya sebentar, Tuhan tolong kabulkanlah.
Bakit no internet ini? Dahil talagang internet? Ha?
Waktu demi waktu, hari demi hari, sadar ku telah sendiri. Kau telah pergi jauh, tinggalkan diriku. Ternyata aku rindu senyuman yang selalu membungkus hati yang terluka di depan mereka. Tuhan, masih pantaskah aku untuk bersamanya? Karena hati ini inginkannya, tak segampang itu ku mencari penggantimu, tak segampang itu ku menemukan sosok seperti dirimu. Cinta, kau tahu betapa besar cinta yang ku tanamkan padamu. Mengapa kau memilih untuk berpisah? Terlalu besar ku taruh harapan pada dirimu, itu alasanku lama tanpa dirimu. Adakah yang bilang ku akan dapat lebih darimu? Tak mungkin, semua itu tak mudah. Tak segampang itu ku menemukan sosok seperti dirimu. Cinta, kau tahu betapa besar cinta yang ku tanamkan padamu. Mengapa kau memilih untuk berpisah? Sosok seperti dirimu, cinta, kau tahu betapa besar cinta yang ku tanamkan padamu. Mengapa kau memilih untuk berpisah? Kau memilih untuk berpisah. Dari kejauhan tergambar cerita tentang kita, terpisah jarak dan waktu. Ingin ku ungkapkan rindu lewat kata indah, tak cukup untuk diri kau terlalu indah dari sekedar kata. Dunia berhenti sejenak menikmati indahmu. Dan apabila tak bersamamu, pastikan menjalani dunia tak seindah kemarin. Sederhana, tertawamu sudah cukup lengkapi sempurnanya hidup bersamamu. Jika hariku lalui tanpa hawamu, percuma senyumku dengan dia. Dan apabila tak bersamamu, pastikan jalani dunia tak seindah kemarin. Sederhana, tertawamu sudah cukup lengkapi sempurnanya hidup bersamamu. Apabila tak bersamamu, pastikan menjalani dunia tak seindah kemarin. Sederhana, tertawamu sudah cukup lengkapi sempurnanya hidup bersamamu. Kalau harus ku mengingatmu lagi, aku takkan sanggup dengan yang terjadi pada kita. Jika melupakanmu hal yang mudah, ini takkan berat, takkan membuat hatiku lelah. Dalam ku akui aku kalah. Cinta ini pahit dan tak harus ku miliki. Jika aku bisa, ku akan kembali. Ku akan merubah takdir cinta yang ku pilih. Meskipun tak mungkin, walaupun ku mau bawa kamu lewat mesin waktu. Jika aku bisa, ku akan kembali, ku akan berubah takdir cinta yang ku pilih, meskipun tak mungkin, walaupun ku mau
Saturday, July 20.
membawa kamu lewat mesin waktu. Jika aku bisa, ku akan kembali, ku akan berubah takdir cinta yang ku pilih, meskipun tak mungkin, walaupun ku mau membawa kamu. Oh, jika ku bisa, ku akan kembali, ku akan berubah takdir cinta yang ku pilih, meskipun tak mungkin, walaupun ku mau bawa kamu lewat mesin waktu. Kan ku bawa wajahmu, kan ku bawa namamu, ku ingin tidur dan bermimpi malam ini. Di sini di kamar ini, sendiri melintas sepi, kusut masang rambut dan gaun malam, ku tak peduli. Di sana kau berdua, di sini aku yang sendiri. Di sana kau tersenyum, di sini aku yang menangis. Jangankan untuk bertemu, memandang pun saja sudah tak boleh, apalagi menyanyi bersama bagai hari lalu. Jangankan mengirim surat, menitip salam pun sudah tak boleh. Ternyata memang kau tercipta bukan untukku. Oh, kan ku simpan wajahmu, kan ku ukir namamu, kan ku buktikan kesetiaanku padamu. Biarlah di sini sendiri merajut hari-hari, bukankah esok atau lusa salam pun sudah tak boleh. Ternyata memang kau tercipta bukan untukku. Oh, kan ku simpan wajahmu, kan ku ukir namamu, kan ku buktikan kesetiaanku padamu. Nak, bila suatu saat kau dengarkan lagu ini dan aku sudah tak ada lagi di sampingmu, kau akan mengerti mengapa begitu menyebalkannya ku di matamu. Nah, jika saat nanti kau telah hidup sendiri dan dunia ternyata tak seperti harapanmu, ku ada di sini menjadi rumah yang selalu menanti kepulanganmu. Kelak kau kan jadi orang tua seperti aku, yang ingin anakmu bahagia dengan hidupnya. Bila bentakan kecilku patahkan hatimu, lebih keras dari itu dunia akan menghakimimu. Ku bentuk dirimu menjadi engkau hari ini. Kau harus kuat, kau harus hebat, permata hatiku. Nah, kan tiba waktu kau harus tentukan jalanmu, yang mungkin tak searah dan indah di mataku. Apabila terjadi, berjanjilah kau akan selalu menjadi dirimu sendiri. Kelak kau kan jadi orang tua seperti aku, yang ingin anakmu berkuasa atas hidupnya. Bila bentakan kecilku patahkan hatimu, lebih keras dari itu dunia akan menghakimimu. Ku bentuk dirimu menjadi engkau hari ini. Kau harus kuat, kau harus hebat, permata hatiku. pergi dari diriku. Tak sanggup harus hidup tanpamu, karena jauh lebih indah bila kita bersama, seperti yang terjadi kemarin. Kini harus aku lewati langkah demi langkah yang menyepi, membalut luka lagi yang kau tahan hingga kini. Habis sudah nafasku menyebut namamu. Oh, takkan hilang cintaku padamu, takkan hilang walau kau memilih pergi, takkan hilang sampai di ujung waktuku. Mencintaimu tak terganti, takkan hilang cintaku padamu, padamu. Takkan hilang cintaku padamu, takkan hilang walau kau memilih pergi, takkan hilang sampai di ujung waktuku. Takkan hilang cintaku padamu, takkan hilang walau kau memilih pergi, takkan hilang sampai di ujung waktuku. Pernah ku sangka ini terjadi, kisah cinta yang suci ini kau tinggalkan begitu saja. Sekian lamanya kita berdua, ku sangka begitu cepat berlalu. Untuk mencari kesombongan diri, lupa segala yang pernah kau ucapkan. Kau tinggalkan aku, pergilah kasih, kejarlah keinginanmu. Selagi masih ada waktu, jangan hiraukan diriku. semoga tercapai segala keinginanmu. Ku sangka begitu cepat berlalu untuk mencari kesombongan diri, lupa segala yang pernah kau ucapkan. Kau tinggalkan aku, pergilah kasih, kejarlah keinginanmu. Selagi masih ada waktu, jangan hiraukan diriku. Aku rela berpisah demi untuk dirimu, semoga tercapai segala keinginanmu. Pergilah kasih, kejarlah keinginanmu. Selagi masih ada waktu, jangan hiraukan diriku. Aku rela berpisah demi untuk dirimu, semoga tercapai segala keinginanmu. Kejarlah keinginanmu. Selagi masih ada waktu, jangan hiraukan diriku. Aku rela berpisah demi untuk dirimu, semoga tercapai segala keinginanmu. Berdiri memutar waktu, teringat kamu yang dulu ada di sampingku setiap hari, jadi sandaran ternyaman saat ku cinta usai di sini, tapi mungkin inilah jalannya, harus berpisah. Terluka menangis, tapi ku terima semua keputusan yang telah kau buat. Satu yang harus kau tahu, menanti kau untuk kembali. Tinggalkan semua di sini. Ku tahu kau pun sama seperti aku, tak ingin cinta usai di sini, tapi mungkin inilah jalannya, harus berpisah. Berharap suatu saat nanti kau dan aku kan bertemu lagi, seperti yang kau ucapkan sebelum kau tinggalkan aku. Kadang hati berharap ku terbangun dari sebuah mimpi, ini bukan kenyataan. Namun adanya tak ada di sini, kau meninggalkanku karena kesalahanku. Mungkin ingin bertemu masih ada, ingin memeluk masih ada. Sayang kini tak bisa, kau telah memilihnya. Mungkin saat hatiku masih sayang, setelah ku memutus cinta, dan kini ku menyesal. Rindu hanya di dalam hati. Andaikan aku tak tergesa memutuskanmu karena egoku, hati ini tak akan begini. Setiap ingat dirimu, rasanya ingin kembali. Mungkin ingin bertemu masih ada, ingin memeluk masih ada. Sayang kini tak bisa, kau telah memilihnya. Mungkin saat hatiku masih sayang, setelah ku memutus cinta, dan kini ku menyesal. Rindu hanya di dalam hati. Ingin memutar waktu, ingin mengulang lagi. Aku tak bisa berpaling lagi. Begitu juga aku, jauh di relung hati. Sungguh tak mampu cinta. Mungkin ingin bertemu masih ada, ingin memeluk masih ada. Sayang kini tak bisa, kau telah memilihnya. Mungkin saat hatiku masih sayang, selaku memutus cinta. Dan kini ku menyesal, rindu hanya di dalam hati. Na 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 na. Sayang kini tak bisa, kau telah memilihnya. Mungkin saat hatiku masih sayang, selaku memutus cinta. Dan kini ku menyesal, rindu hanya di dalam hati. Rindu hanya di dalam hati. Di hati kau tetap terindah. Selalu di hati. Terlalu indah dilupakan, terlalu sedih dikenangkan. Setelah aku jauh berjalan dan kau tinggalkan, betapa hatiku bersedih, menang kasih dan sayang. Setulus pesanmu kepadaku, engkau kan menang. Andaikan kau datang kembali, jawaban apa yang kan ku beri? Adakah jalan yang kau temui untuk kita kembali lagi? Bersinarlah bulan purnama, indah serta tulus cintanya. Bersinarlah terus sampai nanti lagu ini ku akhiri. Betapa hatiku bersedih, mengenang kasih dan sayang. Setulus pesanmu kepadaku, engkau kan menang. jalan yang kau temui untuk kita kembali lagi. Bersinarlah bulan purnama, seindah serta tulus cintanya. Bersinarlah terus sampai nanti. Aku ini ku akhiri. Andaikan kau datang kembali, jawaban apa yang akan ku beri? Adakah cara yang kau temui untuk kita kembali lagi? Bersinarlah bulan.
Good afternoon, ladies and gentlemen. Welcome to the virtual room of regional workshop on biotechnology for food security and biodiversity conservation. We will start this session at 2 p.m. and so the next session will be led by our moderator, that will be Professor Dr. Bambang Purwantara, Magister of Science PhD from IPB University, from Secretary of Board of Trust IPB University, President of Indonesian Society for Animal Reproductive Biotechnology, President of Indonesian Society for Agriculture Biotechnology, and Professor Bambang Purwantara, also graduate from IPB, Denmark, master and doctoral from Denmark. So good afternoon, Pak Prof. Bambang Purwantara.
Good afternoon, Mbak.
Oke,
Assalamualaikum.
Oke, sehat ya Pak.
Alhamdulillah, udah baik.
So, ladies and gentlemen, the session will be started and led by Professor Bambang Purwantara. So, to Professor Bambang, please, Pak, welcome.
Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Salam sejahtera untuk kita semua. Very good afternoon. Distinguished participants, organizing committee, and ladies and gentlemen, also director and deputy director who is now on the plane flying from Hainan to Jakarta. First of all, I would like to apologize to the speaker, especially Ibu Sofiani Dewi, for moderating the afternoon session online by Zoom due to my health condition, because I just recovered from flu, from a bit disturbance of my health. This session, it can be fortunate, it can be unfortunate, because it would be very short, since only one speaker will be delivering her message. As usual, the afternoon session, I think almost always very challenging. Yeah, after having a good lunch, everybody will tend to be very sleepy. So please be wake up, boost your spirit by a cup of coffee beside your laptop, and follow this very important session. First, I would like to start with the rule. The rule is something like this, because we have a 1 hour total presentation and discussion. I would like just to split into equal amounts of time. So the presentation of Ibu Sofiani Dewi will be 30 minutes as planned before. And then we will continue with discussion for more or less 30 minutes. Then we will finish our afternoon session. I would like to start with the curriculum vitae of our speaker this afternoon, Ibu Sofiani Dewi, STP MSI, born in Cimahi. It's 14 hours. The city just beside Bandung. 20 February 1976, yeah. And now she's working with Food and Drug Administration Office in Jakarta, BPOM. And the position now, she is as a food regulator. So related very much with the title of her presentation this afternoon. And Ibu Sofiani graduated from IPB, from the Faculty of Agriculture Technology, mastering on Food and Nutrition Technology, ilmu teknologi pangan, ya. And then she got also master degree from IPB University from food science. For majoring on food science. She has several experience and achievement, especially the position during her tenure at Badan POM. She is head of section of GM and irradiated food standardization. She is also head of the section of certain process standardization, including GM food, irradiated food, organically processed food. And then just recently, during pandemic, she appointed as sub-coordinator for certain process standardization, and then coordinator for standardization of nutrition claim and labeling food for special dietary uses and certain process. And just not far from now, she's a team leader for assessment of nutrition claim and labeling food for special dietary uses and certain processes. For Bu Sofiani, time is yours. Manage your time, and I would like to remind you more or less 2 minutes before end of the session. Please, Bu.
Okay, thank you, Prof. Bambang Purwantara, for the introduction. Actually, I just know that only me speaker in this session. Okay. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
I will share screen first. Good afternoon, ladies and gentlemen. The Honorable Prof. Bambang Purwantara as moderator from IPB University, and also for all participants of this workshop. It is an honor for me to meet you all here to share about regulation and policies on biotechnology product related to food safety, study case in Indonesia. Okay, this is the outline of my presentation. There are legal basis, GM food safety assessment, and the third from GM food control, and the review of the regulations. From the first part is legal basis of genetically modified food. Okay, in the previous presentation from Mbak Ilmi in the morning session, it has been explained about the all regulation about the GM food, all of GM product. So right now I would like to share about the special for food. There is a law number 18 year to 2012 on food. This is the an umbrella of the regulation. And then there is a government regulation number 1886 year 2019 on food safety. And from this government regulation, Indonesia FDA established the regulation number 6 year 2018 on genetically modified food control. And right now this regulation is under review. But beside this regulation, also we are considering about other regulation like law number 6 year 2023 on job creation. Some articles and in food law, it has been changed by this omnibus law, including about the GM food, but it's especially for the licensing in Indonesia for business licensing, perizinan berusaha. So this is our regulation. Just I highlight about the law number 18 year 2012, and then the technically how the GM food control and the food safety assessment, it has been regulated in FDA, Indonesia FDA regulation. From all the regulations, we can conclude that GM food policy in Indonesia, that a safety assessment shall be carried out before GM food is marketed in Indonesia. This is as a precautionary approach principles. So we have to make sure first about the safety before consume by Indonesian people. About the GM food, we need to state, it is stated, clearly stated what about the scope of genetically modified food. In the law and the government regulation, and also in Indonesia FDA regulation, it is stated that genetically modified food are food that are produced or use raw materials, food additives, and or materials produced from the genetically modified products. So there are 4 types of coverage of the definition GM food. So is that fresh food like soybean, like grain, and then raw material like a food additive, and then there is a processing aid like food enzyme that using in the food production, and also processed food that using GM food as an ingredient. Now we are talking about the food safety assessment for genetically modified food. For the safety assessment, in Indonesia FDA regulation, it is there are two materials, main materials that assess assess when the food safety assessment conducted. For the first is the about the genetic information. The genetic information, including the description of the recombinant DNA, the description of host and its use as food, and then the description of donor organism, the description of genetic modification, and characterization of the genetic modification. And the second materials is about the food safety information. It is consists of substantial equivalence and then nutritional modifications, allergenicity, toxicity, and other consideration. These provisions we are use Codex guideline as one of our benchmark in develop this material of food safety assessments. The name guideline for the conduct of food safety assessment of food derived from recombinant DNA plants. For the genetic information, GM food must be able to provide clear information about the identity of the GMOs, the techniques and objective of GMO modification, the nature of GMOs to be assessed for safety, host description, and the history of usage as food. And for food safety information for substantial equivalence, we do the assessment of nutritional and non-nutritional content and compare with non-GM products. So we want to know about the nutritional content of the GM food and then compare with non-GM products, conventional product. And for the nutrition modification, it just assess when there is a change in nutrition value. Further supporting data is needed to find out the GM food doesn't cause effect on health. Usually, if there is a nutritional modification, there is a change in nutritional value, the proponent should provide the feeding the data like a feeding study. For allergenicity, the assessment of GM, we do the assessment on GM protein, including the homology with allergen proteins, the content of proteins, digestibility, and heat resistance. And then for the toxicity, the assessment is done for GM protein homology with toxin protein and acute toxicity tests. And other considerations, if GM food contain antibiotic resistance marker genes, then the GM food safety assessment should include the safety of genes encoded protein or enzymes. So this is the material that assess when we do the food safety assessment for GM food. How about the flow chart of GM food safety assessment? This is this flow chart comes from government regulation number 21 year 2005. So the applicant or proponent should submit their proposal to Indonesia FDA, and then Indonesia FDA will mandate the Biosafety Commission to do the assessment. Biosafety Commission will assign a biosafety technical team for food safety to do the assessment based on the data or documents submitted by a proponent. The timeline is 56 days. This is a calendar days. And then after biosafety technical team for food safety has been done with the for the assessment, then the report will be submitted to Biosafety Commission to get the public participation that uploaded by Biosafety Clearinghouse in Indonesia BCH website for 60 days. After that, Biosafety Commission will do the meeting for discuss about the result of public hearing atau public participation and the result of the report of biosafety technical team for food safety. If the meeting conclude that the GM food is safe, then Biosafety Commission will give the food safety recommendation to Indonesia FDA, and then head of Indonesia FDA will give the safety approval and submit to the applicant. This is the flow chart. Until now, from 2011 until March 2023, there are 56 GM food products that has received food safety approval. There are soybeans product, corn, wheat, canola seeds, cotton seed, sugarcane, potato, and other raw materials. This is a highly refined product. There are two products, are ice structured protein and oligosaccharide, fucosilactose oligosaccharide. The list of GM food that has received food safety approval can be found first in Indonesia BCH, like Mbak Ilmi share in the morning session, and also the list can be accessed in our subsite sandarpangan.pom.go.id. This is for the examples of the information in our website. There is a commodity, event, the characteristic and the gene, promoter, terminator, and the degree of food Indonesia FDA. Now, after the proponent get the GM food safety approval, there are obligations for them that they should submit the GM food samples, and then the control food samples, the counterparts, and then the documents in the form of validated detection method, primary sequence information, and information on where to get the benchmark or certified reference material, if any. This is hopefully with this provision, our laboratorium in FDA, Indonesia FDA, can develop the method for detect all of the GM food that has received the approval or the certificate, food safety certificate. And the other provision is about the labeling. GM food that are marketed in retail packaging shall declare the information on GM on the label using the word in Indonesia in bahasa produk rekayasa genetik. It's mean GM product. GM product in like this. The obligation to declare GM information on the label is carried out for GM food. Sorry, GM food containing at least 5% GM DNA content. So if some product contain less than 5% GM DNA content, so in the product not need to declare about the GM information, but this information is excluded for highly refined products such as vegetable oil, sugar, starch, because in these products it must be there is no DNA content anymore. Now we are going share about the food safety control. The food safety control is for GM food is the same like other prepackaged food. So we have two mechanisms to control the products. There is a pre-market control. It's like audit in GMP or ACCP requirements for food industry facilities, and we do the evaluation on safety, quality, and nutrition of food product. Then the result is the number of for marketing MD or ML, and there's some number 12 digit of number. And the post-market control, we are do the sampling and testing for prepackaged food in the market, and then the control the fulfillment of the regulations, including label and advertisement. Now, just I mentioned previously that we are reviewing the regulation on genetically modified food. We have a management system on food product standardization, like this evaluation and then development, establishment, socialization, implementation, and monitoring. And this like a cycle. Then now our regulation on genetically modified food control based on this management system is under review. The background of the revise or review of the regulation is the development of science and technology, like Pak Romi said in the first session, that science and technology going fast, advance quickly gitu. And then we do the harmonization with the new regulation and the review of the implementation of existing regulation. This is in order to protect consumer health, create fair trade practices, and facilitate innovation. This is there are the materials of the revision, the requirement. The first, the requirements for assessment of stack event. and the mechanism for assessment of genome editing products, and then the requirements of highly refined products that are produced from GM food, and then the requirements of products that are produced from GM microorganism, and the obligation of proponent when their GM food proven cause negative impact to human health. For the first material about the stake event, we do the provisions that each single event shall have food safety approval from Indonesia FDA. This is the first provisions. And the new one, the newest regulation in the revision, but not finished yet, that we adopt the high cover law concept for food safety approval. About the second material is about the mechanism for assessment of genome editing products, that this is the newest technology. For GE products, this should be assessed first by mechanism that established by Biosafety Commission. So proponent will submit the proposal to Biosafety Commission to assess is it the product, is their product is GM or not GM. If the GE product that established as GM by Biosafety Commission, that means that the GE product should be assessed to get food safety approval by following the mechanism that present previously. Third, the requirement of highly refined products that are produced from GM food. There is the obligation of GM food safety assessment is excluded for non-protein based highly refined product and not contain DNA or GM protein. These products such as vegetable oils, sugar, and starch. For the fourth, the requirement of products that are produced from GM microorganism. These products should be assessed for the safety of GM microorganism that has been used, and the final product should be free from GM microorganism materials, including the DNA. So for this product, there are two kind of assessment. First, for the safety of the GM microorganisms and the final product. And then the last is about the obligation of proponent when their GM food proven cause negative impact to human health. What happened if after marketed in Indonesia, and then based on the control, it is found that their product is caused a negative impact to human health. So the food safety certificate will be revoked, and the proponent obligated to take control and prevention measures as well as withdraw the GM food from market. In summary, that regulation and policy on biotechnology product Indonesia has been developed for GM food. Before marketed, GM food shall be assessed to get food safety approval from Indonesia FDA as implementation of precautionary approach principles. Food safety assessment of GM food is carried out based on studies of genetic information and food safety information, including the substantial equivalence, allergenicity, toxicity, and other considerations. Review on GM food regulation and policy should be done in order to protect consumer health, create fair trade practices, and facilitate innovation. Thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh. Yeah, Prof. Bambang. I have been done with my presentation, and I give it back to you, Mr. Prof. Bambang.
Alhamdulillah. Dari tadi kena cekal masih. Saya belum bisa masuk untuk berkomentar. Terima kasih Bu Sofiani. Saya kira, I'm sorry, I should back to English. You saved about 5 minutes earlier to finish your presentation, meaning that we have enough time for discussion. I have not see any. Okay, Pak Saiful already raised hand. No, Pak Saiful, you write down your question through chat box. Do you want to speak directly from your place there? I think better to have direct communication by yourself. Pak Saiful, please.
Okay, thank you very much, Prof. Bambang, and good afternoon, everyone. Thank you very much, Bu Anissa, for the presentation. Is there any, my question about the genetic modified products such as the wheat or corn resistant to glyphosate? Is there any concern from the Commission or the Biosafety Commission or the FDA of Indonesia about the impact with the ecological or environmental approach? I mean, when the products are resistant to herbicide, so the farmer will not have any or less consideration about the weeds because they just need to spray and spray again about the weeds, and without any, it's not harmful to the products. But in the environment, with the very long application, years to years in the field, nowadays there are so many researcher report about the weed resistant to herbicide. I mean, is there any consideration about the another impact in the environment or ecosystem? Thank you.
Before Bu Sofiani answering the question, I think the glyphosate effect is more on environment, ya Pak Saiful, while Bu Sofiani is more on food policy. But let's Bu Sofi respond to your question. Silakan Bu Sofi.
Thank you, Prof. Bambang. Thank you, Pak Saiful, for the questions. Maybe Pak Bambang can can answer. Like Pak Bambang said, that this issue it should be assessed in environment biosafety technical team, ya Pak Bambang ya.
Yeah.
And maybe with Biosafety Commission too, maybe in our discuss about our when discussing in pleno meeting, because this issue has been raised in some event, Prof. Bambang.
Yeah.
But from biosafety technical team cannot do or assess about this because in our guideline that refer to codex in food safety didn't consider about this issue. But this is a good issue, ya Pak Bambang, to be discussed more detail. Thank you, Pak.
Yes, Pak Samsul, I think maybe later on the assessment for feed or food would be related with it, because maybe after spraying the glyphosate in a plant, then we can calculate how much will be retaining in say corn or other part of a plant that use glyphosate for anti what do you call it, wheat product. But I think we can reserve this for tomorrow's session, because we will have a discussion tomorrow, and Pak Samsul can bring the issue again tomorrow morning if there are still some questioning. But for this afternoon session, I think maybe Bu Sophie is not in a position to respond directly, because it's more on product itself, and the grain itself, or the product which is come from grain, GM grain or something like that. Next is Pak Supri, our senior citizen of Biotrop. Pak Pri, can you just directly respond to Ibu Zobi? Monggo Pak Pri. Is it Pak Pri available there? If not, then I would like just to read. Do BP.
Okay.
Yeah.
Okay. Okay, Pak.
Pak Pri. Pak
Yes.
kabar? Sehat ya?
Alhamdulillah sehat. Okay. So my question is because we imported a lot of the grain from USA or other countries, and most of them, I know it is a genetically modified organism, and all imported soybean coming from other countries or USA especially, because we use it a lot, the soybean in the form of tempe and other tahu, something like that. Yeah? Is there any check from Indonesian FDA in this matter? And maybe the second question, how many research institutions in Indonesia registering their GMO product until now, especially in the food? Yeah. Thank you.
Thank you, Pak Pri. I think a question of many people as well, that is there any thorough checking of all of the product which is come from soya from the US, because not all are maybe registered as the producer sending to Badan POM. Please, Bu Sophie, respond to the Pak Pri question.
Okay, thank you, Pak Suprianto ya.
Yeah.
Yeah. About Pak Suprianto's questions, that actually for the entry the soybean. As we know, the soybean is fresh food, and when the import the fresh food import product in the border, FDA doesn't have authority to check in the borderline, in the border area. So there are in the border area there is Ministry of Agriculture, and now Barantan quarantine agriculture quarantine. Yeah. So for the fresh food, the soybean, BPOM cannot check, but there is another institution that do the control. And now we are coordinating with Barantan quarantine agriculture quarantine. Oh yeah, Quarantine Agriculture Agency is coordinating with us, BPOM, to check that to make sure that all the soybean come into Indonesia exported to Indonesia, exported to Indonesia is only soybean GM that has received the safety approval from BPOM.
Thank you, Musafi. I think the importer always ask the the approval from the Badan POM through, of course, to the system by asking the KKH to evaluate. And then from KKH goes down to the TTKH, then finally we can get the approval. So this is the responsibility of importer to report all of the soybean marketed to Indonesia. But again, as Pak Supriyanto, Pak Pri mentioning, I think in the US, the says soya, for instance, it's put in a silo, which is sometimes it's a mix between many different breed or whatever, come from different farmers. And then I hope the importer already bring the document or all of the soybean which is imported to Indonesia. So this is the maximum point that Indonesia can protect ourselves, Pak Pri.
Okay,
but of course, the leaking can be happen to any kind in the importing process. But I think Balai Karantina Pertanian, I think they have the mechanism to check to any product, because for tempe tahu, I think there is almost 95% is the GM. So no question that our tempe and our tahu is is made of a GM soya, and also maybe part of the maize for poultry feed is also made of GM maize.
Yeah,
Okay.
Pak, as I know that Kelantan just
Thank you for your insight, Pak Pri, and I think it's better for us to discuss again tomorrow how this issue will be responded by other narasumber,
other speakers.
Speaker tomorrow.
Yeah, okay.
Again, I see the one raised hand, Bu Sri Ismawati. Mbak Sri, please. Mbak Sri still mute. Please, you're unmute. You unmute your. Yes.
Okay, thank you, Prof. Bambang. Yes, I am interested in following up the question from Dr. Supriyanto, because I am also interested in knowing whether some processed food like sugar, starch are made by genetically modified or engineered wheat or sugar cane or something like that. And if so, since it is already processed, is the DNA contained in the original sugar cane and wheat can affect humans or animals' health? And then my second question, and it's not a question actually, it's a suggestion, because this morning up to this afternoon and maybe tomorrow, we are talking about health impacts from the genetically modified organisms or genetically engineered food or feed. And I was wondering if in the next workshop, probably we can invite medical practitioners that are dealing with genetically caused illness or diseases either on animal or human, something like that. So we as a general public would know the importance of talking about genetically modified or engineered food or feed. So we are more careful in reading the labels. And I think what Bu Sofiani has tried to emphasize is the importance of labeling the genetically engineered food. And that's why we are educated to always read the labels of any kind of food sold in the market or supermarket or anything that we are buying and then we are going to eat, and including to our pets and probably in our chickens, our beef, and so on. Okay, thank you, Prof. Bambang.
Thank you, Mbak Sri. I think Bu Sophie already mentioned about the level, the maximum level of GM content. I mean, it can be labeled, the 5% is the maximum. If below then, there are no need to put any labeling. But please, Bu Sophie, you respond to Mbak Sri Ismawati's question, please.
Thank you, Prof. Bambang. Thank you, Bu Sri Irmawati, for the question. For the first question about, if I'm not mistaken, yeah, just about the DNA, GM DNA. Is it if we consume the GM DNA can be affect to human health? Is it, am I right? For the first question. As in my I shared in my presentation previously that when the GM product is concluded that it is as safe as the conventional product, it means that the GM the safety of GM food is the same as with the safety of the conventional food itself. So there is no risk anymore when we consume the food, including when in the food is still contain the GM DNA, like Prof. Prof. Antonius explained in the first session that actually there's no concern about the DNA, but like the assessment, we are focused in the protein, GM protein that can cause toxicity, can cause toxic or allergen, because this is the new protein that there is, and this protein, the protein that has been expressed by the inserted the new gene by engineered DNA engineered. So we are focused to the protein, GM protein, this protein GM. So when the assessment said that it is safe, that's the product is the safe, like the convenience itself. Like as we know that soybean is the source of the allergen, right? Yeah, jadi soybean is the source of allergen. When we said that the GM GM soybean is safe, it means that soybean, like the nature, is still the allergen, but it's the same as the conventional product. That's for the first question. And for the labeling, Prof. Bambang mentioned first that for the labeling, like the general, the purpose of the labeling itself, that the labeling is to provide the information to consumer about what is the content of the food that is consumed by the consumer, including the GM statement. But for the GM statement, there is a threshold limit when the label of GM information it should be declared on label. The threshold limit is 5% for DNA GM DNA content. That's all, Prof. Bambang. Bu Sri, I hope my answer can.
Yes.
Enough.
Yes, thank you, Bu Sophie. I think
Thank you,
on
Bu.
the chat box, I think I see Bu Evelyn write down the question, but as long as Bu Evelyn is still here, please, Bu Evelyn, you can just directly comment to what Ibu Sophie have in her presentation. Bu
Thank
Evelyn,
you
please.
very much. Yes, thank you very much, Pak Bambang. So my question actually, I'm just curious. So thank you, Ibu Sophie, for your presentation. So my question actually is about the use of GE products. So as I listen to your presentation, the use of those kind of products go through thorough process. So does that mean that the Indonesians do not have to worry about health issues when they take in the GE products? Because I just would like to inquire, because I've heard, if I heard it right, there was a time that there was a health scare for animal products from GE products. So, well, I mean, is there a difference in the use of genetic engineering for plants and animals? The reason for my question was that when GE products from plants, there was no like maybe less health scare, but when it came, or like we eat sweet corn, durian, the spineless durians, but when it happened to genetic engineering of the chicken, for example, the chicken that lays egg without the rooster intervening, only through the feeds, the layer feeds. So there was a little bit of scare, like, oh, do we have to eat the egg that is laid because or through the feeds? But we didn't question about eating the sweet corn and the spineless durian and the other GE products from plants. So the main question is there a difference in the genetic engineering for plant products and animal products? Yes, Ibu, thank you.
Yes, please, Bu Sophie. Question is about the difference between GE or GM, plant GM or GE, or animal GM or GE. So
Right.
it seems it's more worry at people or scaring people when they consume the animal GM product. Any explanation about it?
Thank you, Prof. Bambang. Thank you, Miss Evelyn. This is a difficult question for me. Yeah, because like in my presentation, that actually right now we are still concerned, or we use the codex for the food safety assessments for plant. Yeah, for plant we so if you ask me about is there any difference between the GE plant and animal or GE animal products. Actually, right now about the if the products it is proposed to be consumed, that it's mean that the product should be have safety approval and will be assessed with our guidelines. Uh, and the focus is just the same about the toxicity, allergenicity, substantial equivalence, and other considerations. And maybe I can't give you more detail about what is the difference between both of them. I'm so sorry, Miss Evelyn.
It's okay, Bo. So maybe bring the question later, yeah, tomorrow or.
Well, Flynn, I think both plant and animal GM are equally safe if they already passed the evaluation thoroughly. So don't worry about that, because we one day will have to enjoy the GM salmon already been in the market now in the US. And as long as we know that the processes are according to the right process, then we don't worry about that at all. We eat soya through tempe and tahu for many years, and no one get the effect of the problem with the health problem with those very famous and important food of Indonesia. I see Pak Prima Gandhi from Tokyo. Please, Pak Prima.
Yeah, thank you for the chance, Professor. First of all, I want to introduce myself first. My name is Prima Gandhi. I'm lecturer in Bogor IPB University and now take PhD at Tokyo University of Agriculture. Yeah, I have a question about the differences of import policies, maybe between GMOs of human consumption, like soya, soya bean, and the GMOs for industrial raw materials, such as rubber, palm oil, maybe, such as rubber in Indonesia, and which one is easier about the policy import policies? Okay, thank you.
You mean rubber for industry? I mean non-food, yeah?
Yeah, non-food, rubber for tire, like that.
Yeah. Okay, Bu Sophie, please.
Thank you, Prof. Bambang, Pak Prima Gandhi. About the import policies of maybe some for food purposes and between food and non-food. Is it right? Yeah. Which one is easier? Yeah. About the import policies, all I know is about food. So I can I don't know actually about the difference between food and non-food. But for food, I think because food it is really related to safety, to health. So for food that that imported to Indonesia, there are several regulations that should be apa? Yeah, should be followed, should be fulfilled by industry or business actor, like for the safety. I mean, like soya or food product. There is in food law, our food law. It is very tight. Yeah. So when food will come to Indonesia market, it should be have approval first. For the fresh food, like maybe GM food, like a soybean or any kind of grain like maize, it is must have a safety approval. And for the food product like a beverage, soya beverage, or maybe yeah, another food product by GM. So it is the product should be have approval too. Yeah, like in my presentation about the control, that we have two type of control. That's pre-market control before marketed, and then after marketed, it will be controlled too in post-market control. So the control is very tight. I think, yeah, with other product that non-food, but I cannot explain you about more about the non-food. Thank you.
Yes,
Okay,
thank you,
thank you.
Pak Bima. I
Yeah,
think,
thank you, brother.
frankly speaking, all of the GM product, especially for food and feed, it's much a longer process to be approved. So the importers sometimes frustrated how to get into Indonesia because of the regulation. But I think if for the sake of our safety, so hopefully the process will be shorter now. If compared to the other food, of course, the GM product is also much longer process. But again, BP Badan POM already worked very fast, and not all are tested, but can be also evaluated on DES, Sophie. So we do not need to to make another evaluation when in other countries already been thoroughly evaluated. So that's why we can just have some soybean quite quickly, because in some cases we have a shorten of soybean for tempe tahu very quickly. In only 1 month, we need to import from Brazil, from Argentina, from America. So that's why Badan POM already in place to regulate all of it. Time is off now because we are already 1 hour have our session need to be finished. I would like to wrap up in some point that we can conclude that Badan POM already worked very much on the policy and regulation formulation, and bring the regulation and policy in place. So our big applaud and maybe thankful to Badan POM, who already worked very hard on trying to assess the GM quite quickly, because if we look at the number of GM food approved in Indonesia, I think much more easier and longer in the list compared to the GM which is going to be planted. Before going to the market, I think food safety approval from FDA or Badan POM as implementation of precautionary approach principle is always in place, and for the people, for the Indonesian people, not necessarily worry about consuming the GM based food. Then FDA, Indonesian FDA, also on a process of reviewing some GM food regulation, which we know that there are a lot of new technologies like genome editing, and then Ibu Sophie mentioning about stack gene. Stack gene is meaning that combination of many gene in one plant. So it can be higher production, can be high quality product. It can be retained to or resistant to any bug or diseases, and put it those are 2 or 3 or 4 gene in one plant. It called stack gene plant. So this is also a part of the regulation which is not already available in the old one. So I think Badan POM or FDA already take the initiative on how to make the regulation comply with every part of the Cartagena protocols. I think I would like to end this session by thanking Ibu Sofiani Dewi for your presentation, which is enlightened us with many knowledge, and hopefully some of the question can be bring together into our next session tomorrow for the discussion session, which is maybe interested for all of you to come and join us again. Thank you very much, and I would like to end this session by saying Alhamdulillahirabbil alamin. Assalamualaikum warahmatullahi wabarakatuh. Time back to Mas
Waalaikumsalam
Robi.
warahmatullahi wabarakatuh. Maybe we can give applause for our speakers and moderators today. Thank you very much, Prof. Bambang Purwantara, for leading this session very well. So we have a certificate for our speakers and our moderators. So please, the committee to share the certificate virtual. So thank you for all the participants who joined this Thursday. Yeah. So we will see you on the next day. For tomorrow at 9 a.m. We will have a break room. We will have 2 break room, and we will start tomorrow at 9 a.m. And tomorrow we have 2 break room with the title Harmonization of Regulation and Policies in Developing Biotechnology Product in South Asia, and Regulation and Policies on Biotechnology Product Related to Food Safety Study Case in Indonesia. But before that, we will serve the certificate for our speakers. So for the Bu Sofia, so please welcome to the stage, Ibu. Please welcome to the stage for present the certificate from our manager to Bu Sri Widayanti. Please welcome to the stage, Bu. Okay, thank you. So once again, ladies and gentlemen, thank you very much, and we really thank you for Prof. Bambang Purwantara and all the speakers, ladies and gentlemen. So we will end this session on today, and we will see you tomorrow at 9 a.m. So I'm Alia, and wassalamualaikum warahmatullahi wabarakatuh. Terima kasih Pak Bambang.
Thank
Sehat
you very much
selalu.
for
Terima kasih.
everyone.